आ जाएगी कहीं जब good morning. So, the today's session of the Gian program is going to be started just now by the one tutorial today. So, the tutorial is basically on the methodology related to the gas chromatography and mass spectrometry. So, basic things which are the principles related to this particular machine will be just spread here, and after that you will go into the laboratory where you will see that how this machine works. So, first we have that what are the chromatography, that it is basically a Greek chroma meaning color and graphene meaning that is the writing. Then we have the chromatography is basically a way to separate different parts of a mixture on the basis of their what are the factors that is responsible. One is that the charge, then shape and size, polarity, and that the affinity. So, actually it was earlier planned that the GCMS was described by the Professor Andrzej Bajguz, but he told that the machine he was using is different than we have the machine. So, that is why I am just giving the basic idea about this lecture. Okay. So, we have a Russian botanist that Mikhail Tush in 1903 who has developed the chromatography itself. Then these are the some of the basic principle of the chromatography, that what are the things are under this, that the chromatography works based on the concept of the distribution coefficient that is known as a KD, which explains how a substance that called to analyte is spread between two different phases, that is the stationary phase as well as that of the mobile phase. These two phases can be a liquid, solid or gases that don't mix, and the KD values tell us how much of the substance goes into the each phase. It might be a stationary phase, it might be a mobile phase. So, the higher the KD, that is the coefficient distribution, the more substance will stay in that particular phase and the other. We have two phases that I have already told, stationary and the mobile. This phase is basically stay in place like a solid or liquid that is fixed at one place. Then mobile, this is the phase that moves like a gas or liquid which carries the analyst for the stationary phase. Just have a look at how the first chromatography experiment was conducted by the Toesch. He has just taken the leaf, macerate it, and then have a liquid, that is the sample pigment are there. Here it is the first, that this is your gas column, and on this side the mobile phase, or you can say the liquid phase. In between these we have, that is the calcium carbonate bead. These are the calcium carbonate bead, which is basically a stationary phase, and they cannot move from one place to the other place. So, after going through these things has been separated, that is the chlorophyll A, B, keratinite and xanthophyll. In my opinion, this experiment is already you people have done in the BSc third semester plant physiology experiment in the Aligarh Muslim University, Aligarh itself. So, here we have a sample analyte, that how this is going to be interact. It is a mobile phase, that is solid or liquid. These are some of the phases. What are the possibilities in the chromatography is basically that it may be solid liquid and solid liquid, liquid gas or liquid liquid gas. So, stationary phase may be the solid liquid liquid, and the mobile phase varies as. So, these are the three possibilities that can be generated in the particular. We have then the gas chromatography and mass spectrometry. Here in gas chromatography, the mobile phase is basically a carrier gas. and the stationary phase is a solid or the liquid. This is the basic thing. And in the GC, it is basically it is a technique that used to separate and analyze the volatile compounds. And what are the types of GC? That gas solid chromatography, it was earlier used, where analytes adsorb onto the solid phase, and then we have a gas liquid chromatography, where analytes partition between a liquid and gaseous phases are to be separated. Then we have the mobile phase. The mobile phase in gas chromatography is basically a carrier, and it might be a helium, nitrogen, argon, or the hydrogen. These are the some of the gases which has been used as a mobile phase, which transport the analyte through the system. Then we have the stationary phase, that is in the GSC. The stationary phase is basically a solid absorbent, whereas in GSC it is a liquid coating on an inert solid. Then what are the separation principle, on what basis it has been separated? That the compound separate based on their interaction with the stationary phase and their boiling points. Whatever the boiling point, it may be high or low, on that basis it has been separated. Then we have a carrier gas requirement. What is that? It must be dry, oxygen free, and chemically inert to avoid unwanted reaction with the analytes. This is the, then what are the basic component of the GC we have? That one is the carrier gas controller that regulates the gas flow. Then we have an injection port, okay. Then column, detector, and data processor. These are the component of the GC system. Then अभी आप जब जाएंगे वहाँ मशीन देखेंगे तब आपको ये सारी चीज़ें समझ में आ जाएगी, मशीन में एक आपको करके दिखाया जाएगा. Then we have a sample injection. It may be liquid or solid or vaporized in heat. Then GC column, that is the packed column, capillary column. Then we have a temperature control, that it might be different parts, injection port, column, detector. All three are maintained at a particular specific temperature to optimize the separation of that material. Then we have a detection method, that common detectors include flame ionization detector, thermal conductivity detector, and mass spectrometer. Then we have a detection method. So what are the application and advantages are basically for this GCMS is that it is used in food analysis, forensic science, environmental testing, pharmaceutical industries to identify and quantify volatile compounds. In our case, the plant hormone can be analyzed by this particular hormone. Okay? Then what are the advantages we have? That the GC offers high sensitivity, fast analysis, and excellent separation for volatile and thermally stable compounds. Okay. Then application of the sample, आप जो sample देंगे उसका इसके लिए कैसे apply करेंगे? Then that we have the non-polar and low polar compound are easy to analyze. This is the first point. But polar compound may have issues with excessive retention time, poor resolution, and peak tailing. This is the main thing. Then we have a derivatization method like methylation, salinization can help to improve polarity, distribution coefficient, making polar compounds more suitable for GC analysis. So why polar compound is suitable? Only due to that reason जो है. The sample is dissolved in solvent. अब आपका sample है किसमें dissolve करेंगे? So that is your acetone, heptane, or methanol, avoiding chlorinated solvent due to the contamination risk. So these are the some of the other points. This is the working of the GC. We have this is the basically the vacuum function, maintains low pressure, then enhanced sensitivity and the optimized pathway. Just here injector, that how we are going to inject that particular region where the sample is introduced in the GC system, and then it is heated to vaporize the sample and inject it into the column for separation. Then this is the basically the column long tube inside the GC, inside this type of the column, where the sample separate itself and the stationary phase inside the column interact differently with the different components. This is the just we have opened it here, it will not be open in there. So, the oven maintains the control temperature for the column. It can be programmed to increase the temperature. जो उसका program है computer के through, उसके through हम कर सकते हैं. Then we have launched the detector, the mass spectrometer detector, identify and quantify the component separated by the column itself. Just data system software we have. So, whatever the peak we will get a particular data from that particular system. The data system basically records whatever the output from the detector that processing the chromatogram and mass spectra for the analysis itself. It provides information to help identify the component of the sample itself. Okay. This type of the peak we are just having, it might be a peak representation, that is the comparison of the group we can, it particular one group, two group, three and different. Then the toxicity impact we may also have identified. Then a particular compound that specific retention time that might be 299, 174 are likely to be associated with the identification of compound of each group. So, each particular group have a particular retention time on the basis of which a particular hormone or particular compound can be identified very, very easily, and the GCMS analysis, that use of gas chromatography, mass spectrometry basically allows for precise identification and quantification of our compound with peak areas correlating to the concentration of the each compound. So, finally, we have a conclusion that what we will do after going through the GCMS itself, that a powerful analytic tool, it has a metabolic profiling as well as the toxicology and plant health, and their future application is that this particular technique has a vast potential in our plant biochemistry, aiding in crop improvement, sustainable agriculture and environmental monitoring. So, this is the basic thing, and now we by a member of 10 member from this from here to move into the our instrumentation room, where our technical assistant is there, and he will give you an idea that how this particular instrument will work. Okay? So, वहाँ पे जाएगा? रिकॉर्डिंग खाली कर लो, थोड़े से करके रिकॉर्ड कर लो जो है. So, okay, just to go 10 people, first two line.
MS. So next lecture is hormone profiling in plant tissues, will be delivered by Andrzej, and hopefully he will discuss in a very elaborate way how plant hormone could be identified, then profiled, and all the details he will be discussing. So Professor Andrzej.
Thank you very much. Good morning. Good morning, everyone. I would like to continue presenting the last stage detection in phytohormone isolation. The lecture title is Hormone Profiling in Plant Tissue. In this presentation, I would like to explore the critical methods and analytical techniques used in phytohormone profiling. I would like to discuss key detection parameters and address the challenges in hormone quantification. Understanding these processes is essential for advancing plant physiology, plant biology, plant biochemistry, biotechnology, and agricultural research. We know about fundamental role of phytohormones, their biosynthesis, their metabolism, their application. Despite their biological significance, these compounds are typically present extremely low concentration. We know about it, everything, but making the detection and quality challenging. Several advances analytical techniques are development in this purpose, including, of course, HPLC, GCMS, ELISA, LCMS-MS. Each of these methods, techniques, has its advances and limitations. And the choice techniques depends on the type of phytohormone being analyzed, its chemical properties, the complexity of plant matrix. A very simple reason for choosing a techniques, but it's important, is the availability of appropriate equipment. It's important to understand the key parameters that influence of accuracy and the reliability of this analysis. There are sensitivity, specificity, and quantification accuracy. Of course, the precise measurement of hormone levels requires to use internal standards, particularly stable isotope level phytohormones. This standard help correct the for sample preparation, variability, ensuring that the results are reliable and reproducible. By optimizing the parameters, this means sensitivity, specificity, and quantification accuracy, we can minimize errors and enhance the reliability of phytohormone profiling. Why are internal standards so crucial? We're analyzing plant hormones. Various factors such as sample preparation, extraction efficiency, and matrix effects can induce variability. Internal standards, of course, help correct these inconsistencies, ensuring precise and reproducible measurements. What is an internal standard? It's a compound added to the sample before extraction. It's used to account for variation in recovery efficiency during sample processing, is ion suppression in mass spectrometry, and matrix effects which may interfere with hormone detection. For phytohormones, of course, the gold standard in quantification in using isotope label internal standards. These labeled compounds behave chemically identically to the target hormones, but have a distinct mass signature, allowing for accurate correction and quantification. To achieve the highest level of accuracy in phytohormone quantification, isotope labels are widely used. What makes isotope label standards so efficient? Co-evolution with target hormones. So that means that these label hormones behave identically with native phytohormones during chromatography, ensuring accurate quantification. And the next, distinct signals in mass spectrometry. That means that due to the slightly different molecular weights, they can be easily differentiated from endogenous phytohormones in sample. I showed that some example of isotopes. Of course, by integrating this isotope in LC-MS or GC-MS workflows, we can ensure high precision, minimal matrix interference, and reproducive phytohormone quantification. Let's highlight the key advantages of using isotopes in phytohormone quantification. There are matrix efficiency corrections. That means one of the biggest challenges in hormone analysis. In complexity of plant extract, which contain various interfering compounds, isotope standards experience the same extraction and ionization condition as the target hormone, allowing for precise correction of signal, suppression of enhancement in mass spectrometry. The next, improved quantification accuracy by minimizing signal intensity. Isotope help account for simple preparation, variability, ensuring reliable quantification even in highly complex biological metrics. However, what if isotope label standards are unable? In such cases, structurally similar compounds or synthetic analogs can be used as alternative internal standards. While they may provide some level of corrections, they do not fully mimic the behavior of the target hormone, leading to reduced accuracy compared to isotope label internal standards. By utilizing proper internal standards, we can enhance the precision and reproducibility of phytohormones analysis, making it a crucial step in plant hormone research. What's the next important in determining plant hormone concentration? We need to establish a calibration curve. Its curve is essential for converting instrumental responses, such as peak areas or intensities, into actual hormone concentration in a sample. The process of preparing a calibration curve involves several key steps. The first is stock solution, the second, serial dilution, and the third, of course, internal standard addition. By constructing a well-defined calibration curve, we can ensure that our hormone quantification is precise, reproducible, and free from significant analytical errors. The next step is to analyze, then using chromatographic techniques. I said about LC-MS, MS. Professor Shamsul presents GC-MS, MS. The goal is to construct a calibration curve that accurately relates the instrument response to no hormone concentration. Here's how we built and interpreted a calibration curve. Injection into the instrument. The calibration standards are injection into LCMS-MS or GCMS-MS, and the peak areas of the target compost and its internal standard are recorded. On the x-axis represent the no concentration of the native plant hormones. Y-axis represent the response ratio. The ratio of the native hormones peak area to the internal standards peak area. And the next, what is important, regression, of course, regression analysis. It's, we must use specific statistical software. A linear regression model is applied to establish the relationship between concentration and response. The curve should ideally have a high correlation coefficients above 0.99, indicating strong, of course, linearity and reliability. A well-constructed calibration curve is fundamental for accurate hormone quantification. Now, what we have established the calibration curve, we can use it to determine the concentration of plant hormone in unknown sample. The process involves the following steps. Measurements of the sample. The unknown plant sample is processed and injection into LCMS-MS or GCMS system. Instrumental response, peak area or intensity is recorded. Then, comparison with the calibration curve. The response ratio of the endogenous plant hormone is compared with the established calibration curve. And the last, calculation of hormone concentration. The presented formula represent this analysis. The concentration in sample is determined using equation derived from the calibration, of course, curve. However, there are critical conditions to meet. The hormone to internal standard ratio in sample must fall within a linear range of calibration curve. The next, if the ratio falls outside the standard curve range, the quantification may be inaccurate. And the last, by carefully following these steps, we can obtain reliable, reproducible, and biological meaningful hormone quantification results. When using advanced mass spectrometry techniques such as triple, quadruple, or Q-trap, there are several critical factors that must be considered to ensure accurate hormone quantification. The first is instrument resolution limitation. The next, internal standard concentration. And the third is correction factors for minimal mass differences. In the first cases, the resolution of these instruments, such as LCMS triple quad or Q-trap, is typically 1 Dalton, which may not be sufficient to distinguish interfering peaks from the natural isotope cluster of the target hormone. It can lead to false positive or overlapping signals affecting measurements accuracy. About standards concentration, internal standard concentration is said. What about the correction factors for minimal mass differences? Some isotope level standards differ from the endogenous analyte by only 1 or 2 mass unit. In some cases, a correction factor must be applied to compensate for the minor mass overlap between the labeled standard and target hormones. Here are some key quality control parameters. The first, linearity of calibration curve and limit of detection L of D and limit of quantification L of Q. LOD refers the smallest concentration of plant hormone that can be reliably detected by instruments. LOQ is the smallest concentration that can be accurately quantified, which is typical higher than the LOD. Ensuring that the method meets this criteria is essential for high confidence phytohormone profiling. There are more critical aspects must be addressed: recovery rates and reproducibility. Recovery is assessed by speaking no amount of plant hormones and internal standards into plant extract. Detected concentration are then compared to the expected values to evaluate high efficiency the hormones are extracted and measured. A high recovery rate, typically 80 to 120%, indicates a well-optimized extraction and detection methods. To ensure consistent and reliability quantification, calibration curve and sample analysis should be repeated across different days and batches. By implementing these strict quality control measures, we can ensure that hormone quantification is accurate, reproducible, and suitable for biological interpretation. As you see, table shows specific example of plant hormone quantification. By using high precision techniques, LCMS, MS, or GCMS, we can accurately quantify these hormones and analyze their concentration dynamic under different physiological conditions. There are still several challenges that must be addressed to ensure accurate and reliable results. Key challenges in phytohormone detection are matrix effects, low abundance, and chemical diversity. The first, plant extracts are highly complex, containing secondary metabolites, for example, pigments, protein, sugars, and other. Then can affect signal intensity and lead to inaccurate quantification. Effective simple purification techniques, such as solid phase extraction, are essential to reduce these matrix effects. The second, of course, phytohormones are typically present at extremely low concentration, from nanomolar to picomolar levels, requiring high sensitivity detection methods. And the third, phytohormones belong to different chemical classes. For example, auxin, cytokinin, gibberellin, yasmonate, melatonin, or brassinosteroids, each requiring specific extraction and detection strategies. No single method is universally applied, making methods optimization essential for accurate quantification. Addressing these challenges requires carefully design extraction, purification, and detection protocol to ensure that phytohormone profiling remains highly intensive, specific, and reproducible. The most common used methods are HPLC and GSM. Each technique has its strengths and limitation, and the choice between HPLC GCMS depends on the chemical properties of the hormones, sample matrix, and required sensitivity. Of course, our equipment in lab. In hormone analysis, detection is a critical step for determines the sensitivity, specificity of the instrument. Why is LC preferred over GC for plant hormone detection? Unlike GC, LC does not require derivatization, However, there are some exceptions, making it more suitable for analyzing acidic plant hormones such as auxin, abscisic acid, osmonic acid, salicylic acid. LC is highly versatile, allowing coping with multiple detection, including UV, fluorescence, and especially mass spectrometry. In my opinion, the gold standard for detector is mass spectrometry. LC-MS provides the high specificity, sensitivity, and selectivity. Mass detector allows precise quantification of phytohormones at picomolar levels, making it the most powerful detection method for plant hormone profiling. Given its higher sensitivity and broader availability, LC-MS has become the preferred method for quantifying plant hormones. Among the various detection methods available for further hormone detection, LC-MS stands out of the gold standard due to its high sensitivity. How does LC-MS work? The first step is ionization, the next mass selection, and the third step is fragmentation. In the first step, the plant extract is inducible into the mass spectrophotometer, is a mass spectrometer, where the molecules are ionized to create charged particles. And the next step, the second, the ions are separated based on their mass to charge ratio, MZ, using different types of mass analyzers. The last step, the fragmentation, selected ions undergo further fragmentation to produce high specific mass fingerprints, which improve selectivity. There are, of course, some types of mass analyzer in LC-MS. Triple quadruple time of flight, Q-TOF MS, and ion trap MS. Triple quadruple is the most widely used method for targeted phytohormones analysis, offering high sensitivity and quantification accuracy. Quadruple time of flight, Q-TOF MS, allows for untargeted profiling and structural identification of unknown phytohormones. Ion trap MS is used for in deep fragmentation analysis, helping to confirm hormone structures. Why is LC-MS the preferred method? No needed. I would like to summarize. No need for derivatization, unlike GCMS. High sensitivity capable of detection picomolar concentration of hormones. Superior specificity, minimizing interference from plant matrix compounds. By optimizing LC-MS parameters, we can achieve highly accurate and reproducible phytohormone quantification. In LC-MS analysis, one of the most effective techniques for improving sensitivity in phytohormone detection is multiple reaction monitoring, MRM mode, also known as selected reaction monitoring. What is MRM mode? Instead of scanning all possible ions, it focuses on pre-selected transitions, significantly reducing background noise and increasing detection specificity. It works by monitoring the fragmentation of specific precursors ion into the product ion, ensuring that only the target compound is detected. Why this mode crucial for phytohormone detection? Because it's represent high sensitivity, improve specificity, and represent simultaneous detection. How does it work? The precursor ion is selected in the first quadrupole, Q1. The ion undergoes fragmentation in the second quadrupole, and the specific product ion is monitored in the third quadrupole, confirming the hormone's identity. Some, of course, examples. It's some slides present example procedure for phytohormone isolation and their chromatographic profiles. Since they were already available earlier, so I move on the next slide without the detailed discussion. It's a procedure for grinding, extraction, adding internal standard centrifuge and collection supernatant, dry dissolution in our solvent, and then, of course, using SP column and other washing, and obtain purified extract, and then injection into LCMS MS on the column. Before conclusion, the selection of an appropriate chromatographic column is a next crucial step essential for the proper separation of identification of phytohormone. Why? Next, I compare the efficiency of two different chromatographic columns used for the isolation and separation of phytohormones. The first is a kinetic EVO C18 column, and the second is standard C18 column. The key findings from this comparison are intrinsic in the following points. The first, separation efficiency for phytohormones, and the second, peak shape and in sensitivity. First, the kinetic EVO C18 column demonstrated better resolution, meaning that individual phytohormones were more directly separated from each other. The standard C18 column exhibit broader peaks, indicated lower separation efficiency and potential co-evolution of compounds, which can lead to interference in quantification. The second, since phytohormone exists in extremely low concentration, precise separation is crucial for accurate LCMS and mass quantification. For example, the EVO C18 column, due to its improved resolution, is more effective at reducing matrix interference, making it the preferred choice for plant hormone analysis. Choosing the right chromatographic column is essential for achieving high separation efficiency, reproducibility, and accurate quantification of phytohormone. What else is important? Third is, in addition to selecting the appropriate LC column, maintaining high-quality laboratory conditions, as I know is a good laboratory practice, is essential for obtaining accurate phytohormone measurements. The answer, of course, clean lines, no surfactant used for cleaning floors. They can contaminate LC-MS. The second, using contamination-free laboratory consumables, such as pipette types, for example, Eppendorf tubes, or equivalent high quality, microcentrifuge types, or other plastic material involved in a sample preparation and extraction is essential for ensuring accurate results. Conclusion: analytical methods for endogenous plant hormone are essential to study the plant hormone metabolism and signalic responses. Approaches based on LC-MS have become increasingly important quantification methods. Plant extracts are extremely complex and plant hormones are present in trace amount. So the preparation procedures, which I showed yesterday, including sampling, extraction, purification, and sometimes derivatization, are very important to build high-sensitive LC-MS methods. Solution of methanol and isoprene with little water or formic acid are used to extract more plant hormones. Why, for example, acetone is suitable to strigolactons, SPE is then widely used to purify extract. Different mass spectrophotometers coupled with LC are used to develop plant hormone quantification method, in which MRM mode using a triple, quadruple, or Q-trap mass has much higher sensitivity. Simultaneous quantification of phytohormone profiling and related metabolites can help understand the cross-talk within a whole plant hormone network. Simple and efficient methods determining multiple classes of plant hormone based on LC-MS is becoming possible. What about the future perspective? Improving sensitivity is an eternal pursuit and depends on the advancement of various steps from sample preparation through detection. Selective enrichment methods and novel derivatization reagent providing higher MS responses offer particular promises. The next, high throughput methods can improve the efficiency of plant hormone analysis. Increasing the level of automation of sample pretreatment is promising way. Simultaneous multi-hormone sample pretreatment via combination of multiple SPE mechanisms can present all hormonal information from one sample. Of course, we must need to excellent procedures, improvement in MS techniques, enabling simultaneous monitoring of hundreds of MRM mode transition and fast positive negative switch will make simultaneous multi-hormone detection and enable. In situ detection in MS imaging can provide spatial distribution of metabolites and their relative abundance in plants. However, it still remains far from routine of plant hormones because the endogenous level of phytohormones are so low that they cannot be detected without sample cleaning. Thank you very much. After a break, after 5 minutes, I would like to start the next lecture. Yeah, maybe more? Yeah, of course. 11:30, I will start. welcome you on the next my lectures. It's about application of phytohormones in increasing crop productivity in natural and stressful conditions. Over the next hour, I hope, of course, I will explore how phytohormones, this essential plant signaling and important to plant growth and development substances, can be harnessed to improve crop yield, enhance resilience to environmental stresses, abiotic and biotic, of course, and support global food security. With climate change and a rapid population growth placing immense pressure on agriculture understanding and manipulating these natural compounds is more and more important than ever. Agriculture today faces unprecedented challenges. I said about climate change, rapid urbanization, and shifting food consumption patterns are significantly affected global food security. Rising temperatures in the world, unpredictable rainfall, sea degradation, and increasing biotic stresses such as, for example, pests, threaten crop yield worldwide. Additionally, as the global population continues to grow, the demand for food production is reaching critical levels. To address these changes, scientists and agricultural experts are focusing on developing climate-smart crop varieties, crops that are not only high-yielding but also more resilient to environmental stresses. One of the most promising approaches in this pursuit is the application of phytohormones. We know that naturally occurring small organic molecules in plants act as signaling molecules and have a very important fundamental role in plant biology. By understanding how these hormones function and how they can be manipulated, we can enhance crop productivity and resilience of plants, of course. In modern agriculture and plant biology, improving crop yield, productivity and stress resilience is a top priority. Both biotic stresses such as pests and pathogens and abiotic stresses including drought, extreme temperature and soil salinity can lead to substantial crop losses worldwide. As the global population continues to expand, the demand in higher yielding or more resilient crop is increasing. Meet to these challenges, scientists are exploring various strategies, including metabolic engineering of phytohormones. I presented a similar slide on the previous lectures, so I think that I should omit it. Similar like graph which represent the structures of main groups of phytohormones, which of course I presented earlier. Plants do not rely on a single hormone for survival. Instead, they use, we know, a complex network that work together to regulate growth, development, and responses to environmental changes. Phytohormones can have both positive and or negative interactions, allowing plants to finally tune their biological processes. To maintain optimal growth and stress adaptation, plants precisely control hormone levels through biosynthesis, degradation, and inactivation. This regulation ensures that the right hormone is present at the appropriate time and is right placed within the plant. Although phytohormone exists throughout the plant body, their concentration is very special and temporary. For example, hormones involved in growth and development, such as auxin, cytokinins, brassinosteroids, are often found in specific plant tissue where active growth is occurring. By understanding how these hormones are synthesized, transported, and degraded, scientists can develop strategies, special strategies to enhance plant growth and improve stress tolerance and increase, of course, agricultural productivity. Photosynthesis is the very important fundamental of plant growth and productivity, and instinctively all plant hormones play a role in either enhancing or regulating this process. By modulating photosynthesis, phytohormone directly impact biomass accumulation, stress resilience, or overcrop yield. Different phytohormones contribute to this regulation in various ways. For example, auxin, cytokines, gibberellins, brassinosteroids are primarily involved in plant development, promoting plant cell expansion or growth organ, which can enhance photosynthetic capacity. In the next lecture, Professor Hayat presents the detailed regulation of photosynthesis by brassinosteroids. Because this distinct role of phytohormone engineering and quantification can offer a valuable insight into how different tissues function during various development stages and developmental conditions. As we can see, phytohormones play a critical role in development and stress responses. Plant hormone levels are influenced by two major processes. Anabolic pathways, that mean biosynthesis. Catabolic processes, that mean degradation and inactivation, where the plants break down or regulates hormone levels. Of course, both pathways are essential for maintaining hormonal balance. By targeting the genes and enzyme involved in these pathways, we can enhance traits such as increased drought tolerance, faster growth, and improved resistance to diseases. Beyond biosynthesis and degradation of plant hormones, another potential target for improving growth is hormone signal transduction pathways. By modifying the receptors, transcription factors, or regulatory proteins involved in hormone signalings, we can fine-tune plants' responses to environmental conditions. To manipulate phytohormone levels efficiently, scientists have identified specific target genes involved either hormone biosynthesis as presented as green or catabolic presented as in red color. These genes can be modified through genetics engineering or selective breeding to enhance desirable traits in crops. Some key enzymes include, for example, gibberellin 20 oxygenase, gibberellin 3 oxygenase, and gibberellic 2 oxygenase, which regulate the gibberellic metabolism, affected plant height and growth. Another auxin biosynthesis gene, for example TMS1, which influence auxin production, critical for root and shoot development. Cytokinase SK-X cytokinin oxidase dehydrogenase, which control cytokinin degradation, impacting leaf longevity and stress resistance. Another genes, NCED and LOS5, which regulate abscisic acid biosynthesis, are crucial for stress tolerance. And etc., of course, ACC synthase, ACCI oxidase, regulate ethylene production, which influence fruit ripening and stress responses. By selectivity over expressing or silencing these genes, we can successfully create stress resistance, high yielding plant varieties. For example, auxin are among the most important phytohormones, playing a crucial role in cell differentiation, vascular tissue development, root formation, apical dominance, and tropic responses. How dominance we can see outside this building and the front of the Department of Botany, Al-Muslim University. It's a very good example for this apical domination. By manipulating, of course, auxin biosynthesis and signaling, we have been able to enhance plant growth and productivity. One of the key auxin is indole-free acetic acid. IAA genetics engineering has allowed us to modify its biosynthesis and regulation. For example, of course, IAM gene from Pseudomonas syringa has been used to introduce parthenocarpy, leading to the seedless fruit development in crops like tomatoes and grapes, increasing productivity. The next gene, TMS1 from Agrobacterium tumefaciens, were expressed under tuber-specific promoters, has successfully enhanced tuber formation in potatoes, contributing to higher yields. The next key genetics modification include overexpression, the Arabidopsis UC6 gene in potatoes, and the similar genes in poplar resulted in increased drought tolerance and erect plant structure, and reduced, of course, water loss, making these plants, it's mean, for example, potatoes or polar, better suitable for arid environments. The next gene, GH3 gene family responsible for auxin homeostasis, play a significant role balancing auxin levels in plants. Overexpression Oryza sativa GH3.1 in rice and enhance resistance to pathogen. Similar genes from this family Oryza sativa improved cold tolerance by reducing free auxin levels, preventing excessive growth under stress. These examples highlight how fine-tuning auxin levels can help crops adapt to environmental challenges while maintaining productivity. By controlling not only, but in this case, auxin levels, we can optimize root and shoot development, improve nutrient updates, and increase plant resilience to environmental stressors. Beyond auxin biosynthesis and homeostasis, we know that auxin move within the plants, tightly regulated to ensure proper development and stress adaptation. Overexpression on AVP1, vascular H plus pyrophosphatase, enhances auxin transport, leading to increased cell division and overall plant growth. This modification has been particularly used in improving root architecture and enabling plants to absorb water and nutrition more efficiency. In addition to transport, auxin signaling pathways present under opportunity for crop improvement. That means that auxin signaling mechanism involves key elements such as TRR1, AFB, co-receptor, AUX IAA transcriptional receptors, and ARF transcriptional factors. These elements can regulate how plants perceive and respond to auxin, making them potential target for transgenic modification to enhance stress tolerance and, of course, yield stability. Furthermore, auxin mutants offer insights into stress adaptive traits. For example, UC71D mutants exhibit auxin overproduction, leading to enhanced drought tolerance and altered plant morphology. The next mutants shown in rice linked to auxin hydrolyze activity have resulted in increased grain weight and improved crop yield. Cytokinins are another essential phytohormones which play a critical role in differentiation, leaf senescence, nutrient allocation, and other physiological processes. By manipulating cytokinin levels, we can significantly improve plant growth, productivity, and of course resistance to environmental stressors. Key genes involved in cytokinin metabolism have been targeted for genetics manipulation. If, for example, is IPT, isopentenyl transferase, is a crucial enzyme in cytokinin biosynthesis, responsible for promoting cell division and delaying senescence. The next, CKX, on which I provide many studies, cytokinin oxidase dehydrogenase, regulate cytokinin degradation, controlling plant growth and development. UGT catalyzes cytokinin conjugation, affecting its activity and stability in plants. By enhancing cytokinin biosynthesis or reducing its degradation, we can develop crop with delaying aging, improved drought resistance, especially SKX, and higher yields. While cytokinin offer numerous benefits for plant growth, excessive cytokinin production can sometimes lead to unintended effects such as uncontrolled growth. To overcome this challenge, researchers have developed strategies for conditional expression, activating cytokinin biosynthesis only under stress, such as drought of aging. There are two key approaches, and they have been successfully tested. It's PIT expression under the promoters SAG12, which using the senescence associated with SAG12 promoter to drive IPT expression, allows plants to produce auxin only during leaf aging. It leads to delay senescence, prolonged photosynthesis, and improved biomass accumulation. As seen in tobacco and creeping bent grass, it's similar as expression IPT under SARC promoter. The SARC promoter is stress inducible, meaning IPT is activated only under drought conditions. It has been tested in rice, in maize, and peanuts, where it significantly improved water use efficiency, drought tolerance, and the crop yield under water deficient conditions. This advancement showed that by precisely controlling cytokinin expression, we can enhance plant productivity, stress resistance without compromising overall growth and development. One of the most valuable application of cytokinin manipulation in agriculture is its role in delaying leaf senescence, which directly affects photosynthesis, biomass accumulation, and crop yield. We know that cytokinin naturally prevent premature aging by maintaining chlorophyll contents and sustaining photosynthetic activity. Through genetics engineering, scientists have successfully extended the production lifespan of plants. For example, IPT expression under SAG promoter has been shown to delay leaf senescence, leading to the higher biomass improved yield. And the next, of course, which I said earlier, reducing SKX affect results in elevated cytokinin levels. Beyond cytokinin biosynthesis and degradation, another crucial area for improving crop performance is cytokinin signaling. This pathway determines how plants perceive and respond to cytokinin, influencing growth, development, and stress adaptation. In cytokinin signaling pathway consists of three major components: histidine kinase receptor, AHK, and these proteins detect cytokinin and initiate the signaling cascades. The second is histidine transfer proteins, SHP, and these act as messenger, transferring signals from receptor to response regulators. And another, the third, the response regulators, ARR, the type B especially activates genes that enhance growth and stress response, while type A, RRAs, function as negative regulators, fine-tuning the process. By manipulation these signaling components, we can, the first, enhance cytokinin sensitivity, leading to the better growth regulation and yield stability. And the second, modify stress response signaling, improving plant adaptation to drought, salinity, and nutrient limitation. And the third, balance cytokinin interaction with other phytohormones, can optimize developmental processes without unwanted side effects. Studies on cytokinin manipulation has provided promising results. in improving plant productivity and stress resilience across various crop species. Here are some key findings. I said about IPT expression in rice and maize. I said also on SKX silencing barley, which reduce cytokine degradation, has led to higher cytokinin retention, and of course improved growth and increased yield in plants. And overexpression other gene, for example in tobacco, has improved salt stress tolerance, demonstrating cytokines' role in enhancing abiotic stress resistance. These experimental outcomes confirm that cytokinin engineering is a valuable strategy for developing high-yielding and stress resilience crop. The next, I would like to go to another group, gibberellins, which by manipulating their levels, we have successfully enhanced plant growth, also biomass production and stress resilience. For example, these genes involved is one-in-one final step of biosynthesis, gibberellin 20-ox, increasing gibberellin levels and promoting growth and elongation. The next, of course, enhances active gibberellin production, is crucial for seed germination and flowering. The next, GA2 oxidase, is responsible for GA degradation, reducing growth and leading to dwarf phenotypes, which are beneficial for center crop traits. By controlling the expressions of this gene, we can fine-tune plant height, growth rate, and stress adaptation. This, of course, application demonstrates that gibberellins engineering offers great potential for optimizing plant, not only architecture, but improve yield and enhance stress tolerance. Maybe the same. This is another example, use signal transduction. Maybe I would like to go to another hormone. Abscisic acid is often referred to stress hormone because it plays a central role in plant response to abiotic stresses, particularly drought, salinity, and cold stress. Abscisic acid is responsible for regulating several key physiological processes, including the first, stomatal closure, seed dormancy, embryo morphogenesis, and synthesis of protective protein and osmoprotectants. So that means that abscisic acid signals guard cell to close stomata, in stomatal, of course, closure, reducing water loss under drought conditions. In the second case, abscisic acid prevents premature germination, ensuring seeds only sprout when environmental conditions are favorable. In the next case, for example, synthesis of protection protein, this compound help plants withstand dehydration and oxidative stress. Since abscisic acid is directly linked to drought survival, manipulating its biosynthesis and signaling pathways present a valuable strategy for enhancing crop resilience to water-deficient environments. To enhance drought resistance and stress adaptation, the key genes involved in ABA biosynthesis are ZIP, zeaxanthin epoxidase, NCED, that's mean 9-cis-epoxy carotenoid dioxygenase, and LOS5, ABA3, encodes molybdenum cofactor, which is essential for activation abscisic acid aldehyde oxidase, which is in the final step of biosynthesis of this hormone. Of course, by increasing this expression of these genes, we can successfully develop a crop with a higher abscisic acid levels, which results in enhanced drought tolerance, better water use efficiency, and improved survival under extreme conditions, which have in natural in addition to improving drought and salt tolerance, abscisic acid engineering has also been shown to enhance plant recovery after stress exposure. When plants experience extreme conditions such as, for example, drought, the ability to bounce back after rehydration is crucial for maintaining productivity. I show some key findings in this area. Includes petunia plants overexpressing tomato, and SED1 showed complete recovery after 4 days of drought once rehydrated. It's demonstrate ABA, abscisic acid role in post-stress adaptation. Several, of course, findings, strategies, plans with enhanced ABA signaling displayed faster reactions of growth processes after drought stress, ensuring better yield stability. These studies suggest at present that engineering abscisic acid pathways can improve not only resistance to stress, but also the ability of crops to recover quickly, reducing yield losses after adverse environmental conditions. Maybe the same. Oh. I thought about it. Ethylene. Ethylene is a small gaseous, very molecules, but very important. Ethylene influencing several critical physiological processes, including flowering senescence and leaf abscission. Ethylene regulates aging processes, helping plants shed leaves and flower when they are no longer beneficial. And the second, of course, the fruit ripening. It's accelerate the ripening of climacteric fruits such as banana, tomatoes, and apples, making it an essential hormone for agricultural and food industries. Response to biotic and abiotic stress. Ethylene is involved in stress signaling, helping plant adapt to drought, flooding, head, cold, and pathogen attacks. Because of its dual role in both growth promotion and stress responses, ethylene manipulation presents both opportunities and challenges for crop improvement. To optimize ethylene's effects on plant growth and stress adaptation, we can focus it on modify its biosynthesis and signaling pathways. Ethylene biosynthesis is controlled by two key enzymes. ACC synthase is aminocyclopropano-1 carboxylic acid synthase. This is an enzyme catalyzes the first step in ethylene production and is a rate-limiting factor in ethylene biosynthesis. The next is ACC oxidase, convert ACC into ethylene, determining how much of this hormone is available in the plant. In addition, the ethylene signaling pathway includes ethylene receptors. It's ETR1, 2, ERS1, Per2, EIM4. This protein detects ethylene and initiate absence of this hormone. Active genes involved in stress response, growth regulation is EIN3, EIL transcriptional factors. And the next, of course, ethylene response factors, which mediate ethylene effects on gene expression. By targeting these pathways, we can, the first three, reduce ethylene production to delay fruit ripening and extend shelf life. And second, enhance ethylene signaling to improve stress response, making plants more resistant to drought, salinity, and pathogens. And third, modify ethylene sensitivity to balance growth and defense responses. Ethylene engineering has had a significant impact on fruit production, offering, of course, valuable solution for modern agriculture. The key application includes, of course, reducing ethylene production for delay ripening. For example, transcription melons with antisense, ACC oxygenase exhibit slower ripening, improve texture and higher sugar contents, extended the market shelf life. Apples and tomatoes with suppressed ethylene biosynthesis have firmer fruit and better storage potential, reducing food waste. It's very, of course, important too. And the second, enhancing ethylene senescence for stress tolerance. For example, ethylene insensitive maize mutants demonstrate higher grain yield under drought conditions, highlighting its role in improving agricultural resilience. The next show mutants, ACS7 mutants in Arabidopsis, of course it's not a crop plant, showed increased model of plant in biochemistry of molecular sciences. It showed increased tolerance to salt, osmotic and head stress. Another key is microbial application in ethylene regulation. This means rhizobacteria producing ACC, deaminase, break down ethylene precursors, which reduce ethylene levels in plants and improve drought resistance. Summarizing, these findings demonstrate that controlling ethylene biosynthesis and signaling can optimize both food production and stress resilience, making agriculture more suitable. In the next slide, I would like to present challenges and considerations. While ethylene manipulation provides numerous benefits, including improved stress tolerance, enhanced fruit storage, and optimized plant growth, it also presents significant challenges that must be carefully addressed. One of the key issues is ethylene dual role in plant development and stress responses. While reducing ethylene production can delay ripening and improve growth tolerance, excessive suppression may negatively impact normal growth process and weaken the plant's stress resistance against pathogens. On the other hand, increasing ethylene levels can enhance stress resilience, but it may also lead to premature aging and reduced crop yield. To ensure optimal outcomes, precise regulation of ethylene-related genes is, of course, essential. Instead of completely blocking ethylene production, targeted genetics modification using conditional promoters allow ethylene expression only under specific environmental conditions. Another promising strategy is to modify ethylene receptors rather than biosynthesis genes, providing more controlled responses without disturbing overall growth. We know about that brassinosteroids are polyhydroxysteroids, influence cell elongation, cell division, and making them, of course, essential for normal plant physiology. One of the most important roles of brassinosteroids is in cell elongation and division, which contribute to overall plant height, leaf expansion, and root development. They also regulate processes such as embryogenesis, pollen tube growth, and senescence, ensuring that plants undergo proper reproductive development. Brassinosteroids have been found to enhance photosynthesis efficiency by improving chloroplast function and increasing carbon assimilation, ultimately leading to higher biomass production. In addition to their role in normal, of course, in normal conditions, brassinosteroids help plants withstand environmental stress. Under drought conditions, brassinosteroids improve water use efficiency by regulating stomatal conductance. In saline environments, they enhance ion homeostasis, reducing the toxic effect of excess sodium accumulation. In another example, in response to heat and cold stress, they activate protective mechanisms that help maintain cellular integrity. These properties make, of course, brassinosteroids an attractive target to gene manipulation and agricultural application aimed at improving crop resilience and yield stability. To enhance plant growth and stress resilience, we can focus it on modification, biosynthesis, and signaling pathways of brassinosteroids. These hormones are synthesized through serious enzymatic reactions with key genes controlling the production and activity. One of the most, of course, important genes is dwarf 4 dwarf. Thank you. DWAR4, which encodes steroids 24 alpha hydroxylase, a key enzyme in brassinosteroids biosynthesis pathways, which I will show in the next lecture, entitled around the brassinosteroids in algae. Of course, the biosynthesis pathways I show. Overexpression of this gene has been shown to increase plant size, improve yield, and enhance stress tolerance in multiple crop species. In addition to biosynthesis, the brassinosteroid signaling pathways plays a crucial role in determining plant responses to environmental conditions. We know that receptor in this transduction is BRI1, BAC1 receptor complex is at the center of brassinosteroid perception, triggering downstream signaling cascades that regulate gene expression related to growth and stress adaptation. Further downstream, signaling components such as BSK13, BN2 kinase, and transcriptional factors like BZR1 mediate brassinosteroid responses, balancing cell expansion and stress tolerance. Brassinosteroids have been widely studied. Thank you again. Widely studied for their ability to enhance plant growth, productivity, and stress tolerance. By optimizing their biosynthesis and signaling, we have successfully improved. We can successfully improved crop performance in various agricultural settings. One of the most significant application of brassinosteroids is in increasing grain and biomass yield. Studies have shown that overexpression of DWAR4 gene in rice led to 15 to 44% increase in grain yield, while in Arabidopsis as a model plants, the same modification results in larger plants and 59% increase in seed yield. Of course, it's not important. Comparison is maybe enough. It's not necessary. In addition to improving growth, brassinosteroids play a crucial role in helping plants cope with environmental stresses. About heavy metals, I will talk later. Exogenous application of brassinosteroids such as 24-epibrazzinolide or 24-homobrazinolide has been found to enhance photosynthetic efficiency and stringent antioxidant defense system, particularly in tomato and eggplant under heat and salinity stress. By the way, please see the Professor Shamsul Hayat publications in detail, which show. Brassinosteroids have also been shown to mitigate the toxic effects of heavy metals, as said, such as, for example, cadmium in tomato, making them available tools in improving plants' health in polluted soil. Brassinosteroids, foliar spray in cadmium-stressed tomato plants efficiently neutralize heavy metal toxicity and increase fruit yield, proving that this hormone can help plant thrive in a polluted soil. Similarly in rice and the overexpression of exordium, exogene, a brassinosteroid responsive regulator, resulted a large leaf and root system, leading to improved nutrient uptake and higher biomass production. Another breakout involved application of erect leaf rice mutants with altered brassinosteroid signaling, which demonstrate greater light penetration into the, for example, canopy, increasing photosynthesis and boosting grain yield. And this finding shown how brassinosteroids can be manipulated not only to enhance plant growth, but also to improve overall resource use efficiency, making crops more productive and resilient. And these studies confirm that brassinosteroid engineering is a promising approach in modern agriculture. While brassinosteroid enhance plant growth and stress resilience, their application must be carefully controlled. Additionally, brassinosteroids interact with other hormones, for example, auxin and cytokines, meaning that enhanced growth may come at the cost of reduced stress tolerance. To address this challenge, we are exploring target genes modified that regulate biosynthesis, regulate brassinosteroid levels in specific tissue and or developmental stages. Additionally, combining brassinosteroid engineering with other phytohormonal modification, such as gibberellins, for high control or abscisic acid for drought tolerance, can help achieve a more balanced and optimized crop phenotype. By carefully fine-tuning brassinosteroid biosynthesis signaling and interactions with other phytohormones, we can develop high-yield stress resilience crop with maintain a strong structural integrity. The next group which I would like to discuss is yasmonates. Yasmonic acid is a crucial group, is crucial hormone involved in plant defensive stress adaptation and development. They play a central role in responding to wooding, herbivore pathogen attacks, and other environmental stresses such as salinity, drought, and cold. Beyond the role in defensive, yasmonates also regulate seed germination, root growth, tree home formation, flowering, and senescence. This hormone act as signaling molecules that activate protective responses, allowing plant to adapt to challenges and environmental. Due to their role in stress resistance and growth regulation, yasmonates have become a key focus in agricultural biotechnology. By modifying yasmonate pathways, researchers aim to develop a crop with stronger pest resistance, enhanced drought tolerance, and improved overall resilience. To enhance this plant defense, we have focused on modified yasmonate biosynthesis and signaling pathways. Of course, I present the key enzyme in yasmonate production includes 13-lipoxygenase, 13-LOX, which catalyze the first step in yasmonic acid synthesis, and yasmonic acid carboxyl methyl transferase, JMT, which converts yasmonic acid into its active form, as mean methyl yasmonate. In addition to biosynthesis, yasmonate signaling pathways involves CO1 receptors, JAZ receptors, MYC2 transcription factors, which regulate plant response to stress. Of course, the genetic modification targeting these compounds have led to improved resistance to pathogens, enhance drought tolerance, and increase yield stability. By fine-tuning yasmonate biosynthesis and signaling, we can develop crops that are more resistant to pests and environmental stresses while maintaining optimal growth. In the next slide, I would like to show this modification. Maybe this is similar what I said. Yasmonates, maybe it is not you're just interested. Something else. Phytohormones, of course, play a critical role in protected plants against biotic stresses. One direct defense strategy is the production of secondary metabolites such as alkaloids, cyanogenic glycosides, and glycosinolates. This compound act as a natural pesticide, deterring herbivores and inhibiting pathogen growth. Phytohormones such as yasmonate and yasmonic acid regulate the biosynthesis of these defensive compounds, ensuring to release the volatile organic compounds which attack natural enemies of insect pests. The signaling molecule, that's mean yasmonic acid and salicylic acid, guide predatory insect to the prey and enhancing biological pest control. Key volatiles such as farnesan, methyl salicylate, and oxymen have been identified as a major player in this plant-insect communication network. The future of phytohormone research lies in integrating advanced technology to further enhance plant productivity, stress resilience, and sustainability in agriculture. One of the most promising area is plant hormonomics, which combines omics technologies to analyze hormone interaction at the molecular level. It allows scientists to gain deeper insight into how phytohormones regulate growth and stress responses. Another key focus is the development of climate resilience crop through target hormonal manipulation. With climate change posing increasing challenges to food security, so research are engineering crop that can better withstand extreme weather conditions, including drought, heat, and soil salinity. By fine-tuning phytohormone pathways, we can improve water use efficiency, nutrient uptake, and resistance to water environmental conditions. Enhancement in biosensor and omics tool are also revolutionizing phytohormone research. These technologies enable precise hormone profiling, allowing for real-time monitoring of hormone level in plants. It's of course future. It can lead to more effective breeding problems and agricultural practice that optimize hormone regulation for maximum yield and resilience. Finally, Sustainable agricultural strategies are increasingly leveraging hormone-based approaches to reduce resilience on chemical fertilizers and pesticides. By harnessing the natural signaling power of phytohormones, future crop management practice will become more environmentally friendly and efficient. What's the next? Phytohormone as a play a fundamental role in regulating and stress, the ability to enhance crop productivity and resilience makes them invaluable tool for modern agriculture. A comprehensive understanding of phytohormone mechanism will allow research to optimize their application, balancing growth, defense, and adaptation. The integration of genetic engineering, precision agriculture, and omics technology will further expand the potential of phytohormones in creating more effective and sustainable future for agriculture. So thank you very much for your attention.
brassinosteroids. Okay. So just we have a little bit brief introduction about the hormone, as Professor Bajguz has already told you. So there is no need going to be the detail in the introduction, but as everyone is known that the hormone, if it is related to the plant hormone, then it is known as the phytohormone. In short, I can say that there is a well-known 6 plant hormone, that is basically the auxin, gibberellin, cytokinin, ethylene, and the latest one is your brassinosteroids. So, but besides this hormone, we have also some signaling new molecules, as you have already seen in that lecture, that there is a jasmonate, salicylic acid, and strigolactone. So, as you know that it is directly related to that of the brassinosteroids only. So, brassinosteroids are basically a polyhydroxylated sterol derivatives that is present in all plant species. Most of the species you can say, and share the structural similarities with that of the animal and insect steroid hormone. Basically, the BR was discovered through the examination of pollen extracts from over 30 species by Michel and his co-worker in the United States Department of Agriculture in West Valley, USA. The growth promoting extract, which was first isolated from the Brassica napus, and they named it as brassins. So, the most bioactive brassinosteroids are basically the brassinolide, 24 epibrassinolide, as well as that of the 28 homobrassinolide. Out of all these brassinosteroids, these three are the most active brassinosteroids till date. That is one is your this brassinolide, then we have this epibrassinolide 24, and this the 28 homobrassinolide. And out of these brassinolide, the 24 epibrassinolide is most susceptible, or you can say the most active to the plant part. Just have a brief introduction about their role, that how it is going to be increase the plant growth and development. Just see the slide, there is a number of response was generated by the brassinosteroids: stomatal development, seed germination, then we have the regulation of the target, then the cell division and cell wall, then we have the redox, then we have the RCS, and these are all the functions that has also been present in the other hormone, but some of them of the effect of the brassinosteroids are so specific that has been mentioned here. So, we can say that the brassinosteroid cannot be considered long distance signals, as they are incapable of being transported between roots, shoots, or between organs within a shoot. So, on this basis, you can say that it is not a long distance signal. We are basically indirectly contribute to long distance signaling by altering the oxygen transport system. So, it means that the direct effect of BR is not exact, but first it affects the response of the oxygen, which indirectly affect the plant growth and development. So, there is no direct evidence of their response. They regulate the various physiological and morphological process in the plants, including the photosynthesis. So many processes are there, but one is the most important, that is the photosynthesis, which is directly related to that of the crop yield. So, as you know about the photosynthesis, that the photosynthesis is a highly controlled processes, involves capturing solar energy through chlorophyll, exciting electron, transferring, and so on. So, it is not so many things are involved in enhancing or in increasing or marginalizing, or you can say there is so many mechanisms are involved for the photosynthesis itself. So, as you know, the primary sites are the chloroplast for the photosynthesis. Green pigment, chloroplast is a green pigment, and photosynthesis is directly related to that of the chloroplast for the synthesis of the photosynthesis, where both light and dark reactions are occurred. There is a two photosystem, that is the photosystem 1 and photosystem 2 in the thylakoid membrane, and they function separately, not like a same time. The PS1 is found is in as you know that the stroma is a stacked and non-stacked membrane. So PS1 is found in the non-stacked stroma membrane, while PS2 reduced largely in the stacked grana membrane. Whereas the PS1 basically the absorb light maximally at 7 nanometer and PS2 at 680 nanometer. These are the well known thing, there is no need of describing here. So these points are very well common, that so there is known. As you know that there is a extensive data are exist on the effect of brassinosteroids on plant under various abiotic stresses, that is the salt, sorry, that is the salt, then we have the temperature, water, as well as that of the metal stress. So there is a two different theory which suggests that the brassinosteroids may prevent the loss of photosynthetic pigments by inducting or inducing or activating enzyme involved in the chlorophyll biosynthesis. This is the one possibility. Then we have the another possibility that where it is going to be work is that BRs may overcome the stomatal limitations, then increasing the internal carbon dioxide concentration for the various photosynthetic enzymes, and that leading to the enhanced carbon fixation efficiency. And so we have that the maintaining an optimal endogenous hormone level is critical for normal plant growth and development, and that require a balance between the hormone biosynthesis as well as that of the catabolic processes. Brassinoxal, which is basically known inhibitor of the brassinosteroids, and the BR biosynthesis genes such as your DT2, DW4, CFD are upregulated, and in condition with excess brassinoid, the brassinosteroid inactivation gene that is related to your BAS1 is upregulated. This is the main. Then we have that key rate limiting steps in the BR biosynthetic pathway also include the oxidation and hydroxylation at both C23 and C20 level at the steroidal side chain. Then we have the another thing, that is the cytochrome 450 monooxygenase. This is the families, another family, that is the CYP85 and CYP90. They catalyzes their oxidation and hydroxylation reaction. And besides that, the other group, that is the CYP851 and the CYP8582, they catalyzes the C6 oxidation. These genes are, all these genes which are involved are under the negative feedback that may be controlled by the brassinosteroid receptor signaling. And the expression of that particular rice CYP3 in Arabidopsis also due to that of the inactivation of the brassinosteroids. As you know that always there is a receptor in the plasma membrane So, BR1 is the receptor for the brassinosteroids. As some of the students are from the MSc, where we have taught in the MSc that receptors are generally present on the plasma membrane, and for each and every effect there is a particular receptor. So, here we have a receptor that is known as BRAVE1, which is basically a receptor for the brassinosteroids itself. So, brassinosteroids generally play a crucial role in plant growth and development, as already told by the Bajguz and myself, and also that the studies basically involving the brassinosteroid mutant have been pivotal in demonstrating the role of brassinosteroid in the plant hormone. Then there is also some effect that the BR signaling is now considered as one of the most comprehensively studied pathway among the plant hormone. So, as a various signaling pathway is available, but the BR is one of the best signaling pathway. As I have told you that there is a BRAVE1 is the receptor or the gene, here brassinosteroid insensitive is a crucial component of the BR signaling pathway, and BRAVE1 was identified through the experiments on the Arabidopsis, where a mutant with normal root elongation despite high brassinoid concentration were isolated. And all these mutants basically exhibited their developmental deficiencies such as the impaired hypocortical elongation, which I have already told you, and inhibited the primary root growth, which could not be rescued even by the BR treatment. So, brassinoid binds to a BR1 with high specificity, confirming its role as a BR receptor. And this BRAVE1 is a leucine rich repeat receptor, that is serine threonine, and localized is basically in the plasma membrane itself. And another receptor kinase, that is the LRR, form the largest receptor class that is to be predicted in the Arabidopsis genome itself. Then the conserved structure of BRAVE1 that includes your N-terminal extracellular domain, with that is the 25 tandem LRR motif. Then we have a single transmembrane domain, and next one is the cytoplasmic kinase domain that is specific to a particular amino acid, serine and threonine residues. Besides that, the flanking region of LR2 along with the Iceland domain constitute the minimum binding site of brassinosteroids. Now we have the mechanism in relation to that of the brassinosteroids, but as we know that our this topic is directly related to that of the photosynthesis, that how it is going to be regulated. So, now we have to go to on the photomorphogenesis regulation by the brassinosteroids directly. Apart from the mechanism of action is not needed here. So, as the photosynthesis and photomorphogenesis both are the light regulated processes that are heavily influenced by the brassinosteroids, and the photomorphogenesis refers to plant growth and development in the presence of the light. Then we have that the brassinosteroid level are not directly affected by the light, but regulate light signaling components. Then the another point in that the BR repress photoreceptors like the phytochrome B and phototropin, which promote the photomorphogenesis. Brassinosteroid further activate the negative regulators of photomorphogenesis, that is the COP1 and SPA1. So COP1 basically targets the targets and degrades by 5 a key promoter of photomorphogenesis. So brassinosteroid also reduce the expression of GATA2 and GATA4, which promote the photomorphogenesis. Another point is that brassinosteroids are the one hormone which is an essential for the gibberellic acid mediated elongation. So whatever the gibberellic acid mediated elongation is known, it is related to that of the brassinosteroid by allowing the BZR and PFR complex to bind the DNA and promote the etiolation. That is a indirect effect. Then we have the brassinosteroids promote the plant topes and lateral root development by interacting with the auxin or its transporter. Then some of that stomatal development is regulated through this erecta receptor kinase pathway. BIN2, that is the another gene which inhibits the IODA, a mitogen activated protein kinase, and speechless, that is a BHLS factor, which respectively suppress and promote the stomatal development. Then it is going to be much more in detail, so just to short the lecture, that the regulation of photosynthesis now come to the brassinosteroids. So as you know that the several studies have examined the effect of brassinosteroids on the photochemical reaction occurring in the thylakoid membrane by measuring the chlorophyll fluorescence that you will be followed in the afternoon session. Then the Hill reaction activity in the chloroplast was used to assess the electron transport during photosynthesis. And as I have already told that the HBl, that is one of the brassinosteroid, led to decrease in the Hill reaction activity. However, this activity improved later at developmental stages. Then the efficiency of photosystem 2 measures the fraction of the absorbed photons that involved in the photochemistry, which determines the linear transport rate and reflects the overall photosynthetic efficiency. Besides that, the chloroplast also serve as the primary organelle that is responsible for the photosynthesis, a process that is divided into the two key phases. One is that is basically a light absorption phase and another is the carbon fixation phase. As you know that chlorophyll is a key light absorbing pigment where it captures the solar energy to drive the ATP synthesis, and the efficiency of this energy captures directly affect the photosynthetic capacity. The degree of leaf greenness is strongly correlated with chlorophyll content, and a greenery leaf typically indicate the chlorophyll level, which in turn reflects greater photosynthetic potential. It means that if the color of the leaf is much more green, it means that there is a large of chlorophyll in it, and it has a more capability to photosynthetic activity as compared to the yellow leaves, where we have a less chlorophyll content. The measuring chlorophyll level in leaf is one of the most reliable parameter that used to assess the photosynthetic performance of the plant. Higher chlorophyll content generally implies on the improved capacity for solar energy absorption and utilization. Brassinosteroids are known to significantly boost chlorophyll content in plants by increasing the chlorophyll level, and the brassinosteroids also play a crucial role in enhancing the photosynthetic efficiency and overall plant growth. Plant possess two types of chlorophyll, that is your chlorophyll A and B, and in the presence of brassinosteroids, it is reported that the total chlorophyll content significantly increases, enhancing the photosynthetic efficiency. Among the total chlorophyll, carotenoid content is also elevated under the influence of 24 epibratinoids, contributing to improved light capture and photoreception. Brassinosteroids have been shown to boost chlorophyll levels in various plants, including Vigna radiata, Brassica gensea, Cicer aratinum, Oryza sativa, Vicia faba, Triticum aestivum, and Pelargonium. In, in this, Vigna, Brassica, Cicer, Vicia, Triticum, the work was also conducted in our lab. The brassinosteroid enhanced the light harvesting efficiency of plants by promoting the transcription and translation of enzyme that is critical for the chlorophyll biosynthesis. This regulatory role makes them essential for optimizing plant growth and productivity. As you know that there is a increase in the photosynthesis, so there might be involvement of the some enzyme, and the Rubisco, a crucial enzyme in the Calvin cycle, which catalyzes the carboxylation of ribulose bisphosphate and playing a central role in the carbon fixation during photosynthesis. Its activity is regulated by the Rubisco activase, which ensures the proper functioning of the Rubisco. Brassinosteroid enhance the Rubisco activity, contributing to improve the photosynthetic efficiency in plants itself. And the increased activity of Rubisco and other Calvin cycle enzymes in the presence of brassinosteroid is due to the upregulation of a particular genes which is responsible for encoding these enzymes. Brassinosteroids upregulate the genes such as the RBCL, Rubisco large subunit, or RBCS, that is the Rubisco small subunit, and other coding for enzymes involved in the Calvin cycle reaction that include the triose phosphate isomerase enzyme. Then we have the glycerate phosphate 3 kinase and the fructose 1,6 bisphosphate, then pseudoheptulose 1,7 bisphosphate, and the ribulose 5-phosphate kinase. This regulatory role of brassinosteroids highlights the importance in enhancing the carbon fixation on the crop. The net photosynthetic rate and associated attribute show improvement in the presence of brassinosteroids. This increase in photosynthetic efficiency is attributed to elevated levels of carbon dioxide assimilation, enhanced Rubisco activity, and improve the stomatal conductance. And it has also been reported that brassinosteroid improve the net photosynthetic rate in some plants like that of the Limus chinensis. Various physiological parameters such as that of the chlorophyll content, stomatal conductance, internal carbon dioxide concentration, and transpiration rate increases when the plants were sprayed with the HBL or the EBL, which is a two different type of the brassinosteroids. Along with the photosynthetic rate and the chlorophyll content, the total sugar content also increases in the presence of the HBl. Carbonic anhydrase enzyme, that is also that of the like of the Rubisco, its activity increases under the brassinosteroid treatment, where the leaves of the Brassica gentiana and Vigna radiata treated with HBl showed the higher carbonic anhydrase activity. The studies involved the BR treated plant and BR deficient mutant established a direct connection between the brassinosteroids and that the genes involved in the photosynthesis. The research on Arabidopsis mutants with altered BR response revealed a enlarged thylakoid as well as that of the smaller photosynthetic system complex, and also inhibited the evolution of the carbon dioxide and reduced the pH2 quantum yield. And all these findings demonstrate that brassinosteroid enhance the photosynthetic efficiency of photosystem 2 and increases the chlorophyll content, which promote the activity of various Calvin cycle enzymes by upregulating the genes involved and ultimately improve the overall net photosynthetic rate. Then we have a effect of brassinosteroid on photosynthesis in plants in the presence of the various stresses. There is a number of stresses are there, such as that of the salt stress, as well as that of the water stress, heavy metal stress, and so on. So just we have summarized some of the work of the different stress in some of the slide. That the salt stress, basically the soil salinity is a major environmental constraint which basically hampers the overall plant growth and productivity, particularly in our agriculture system. And the salt stress causes the reduction in the photosynthetic pigment as it can break down the chlorophyll. These are the various effects of the salt stress on the various plants. In some of the species like Helianthus anus, Triticum aestivum, and Ricinus communis, the level of the various chlorophyll that of the A, B decrease under the salt stress. But if these salt stress plants are sprayed with the brassinosteroids, the chlorophyll which has been affected is restored by the epibrassinoid or homobrassinoid or brassinoid. For example, in the rice variety, that is the Pusa Basmati 1, chlorophyll content basically decreased under salt stress, but the addition of the EBL helped to restore it. Salt stress typically causes a decline in the photosynthetic rate and relative attribute. However, the treatment of the brassinosteroid can help to restore the damage which has been caused to the photosynthetic machinery under the stress condition. In a study on Vigna radiata seedlings, the salt stress significantly decreased the photosynthetic rate, spared chlorophyll, and the fluorescence. However, when these plants were treated with the brassinosteroid, the damage was partially reversed. The highest photosynthetic value were observed in the seedling treated with the brassinosteroid alone. It means that the decrease caused in the salt stress plant can be overcome by the treatment of the brassinosteroid. A similar result were also observed in the other plant. In a study on Lactuca sativa, that is the various concentration of NaCl reduced the chlorophyll content. However, when the plants were treated with brassinosteroid, all the concentrations successfully alleviate the damaging effect of the salt stress. In the Cicer aratinum, the chlorophyll content, both the A and B, increased when it is treated with the HBl, which has been reduced due to salt stress. There is some other effect of the salt and brassinosteroids are reported in a number of crops. So it is not possible to summarize all the result in one lecture. Then we have the another effect, that is the water stress. So basically the drought stress occurs when there is a availability to plants decrease to a lower that become unfavorable for growth, that is, as you know, that it is called as the water stress. So under the water stress, the photosynthetic rate as well as that of the stomatal conductance as well as that of the internal carbon dioxide concentration was generally decreased. But if these plants were treated with that of the brassinosteroid, so it is reported that this decrease can be overcome and the values can be elevated. Whereas it is reported in the case of the rice, which requires a ample of water. It is the one crop which required much more water for the growth. So drought stress reduces the photosynthetic conductance, then the water use efficiency. However, when we are giving a epibrassinoid as a foliar spray, it improves the overall plant growth. Then we have the brassinosteroid treatment that mitigate the adverse effect of drought on photosynthesis by maintaining, as you can say, the increasing the use of light and non-photochemical quenching in the photosystem too. In barley cultivars with impaired C6 oxidase activity, that showed a lower gene transcript accumulation related to the stress response such as the HS high temperature, that significant, you can say that the high temperature is a major threat to the crop production. So, which cause a damage to mesophyll cell, that is a permanent damage, then increasing the plasma membrane permeability and decreasing the water availability, which disrupt the leaf water potential. As which you have, I think, the on the yesterday tutorial for when measuring a leaf water potential, you may seen that in the stress plant there is a disruption of the leaf water potential. Photosynthesis is highly sensitive to temperature and its efficiency is impacted by the heat stress. AP brassinosteroid application help to reduce the thermotolerance in plants like rape and tomato, rapeseed, and under heat stress EBL protects chlorophyll, maintaining levels of chlorophyll A, B, and total carotenoids. EBL treatment also increases the stomatal conductance, which is directly or indirectly linked to the higher transpiration rate. Then we have a well-known hormone, that is basically a abscisic acid, which is called as a stress hormone. It is also regulate the physiological process and is induced during high temperature stress. So, EBL application enhances the ABA synthesis and helps the plant to tolerate heat stress by promoting the production of the heat shock protein. So, under heat stress, the key photosynthetic parameters like photosynthetic rate, GS, internal carbon dioxide concentration, quantum efficiency of photosystem, and photochemical quenching is decreased. The detrimental effect of heat stress on these parameters are mitigated by the application of the AP brassinosteroid. So, apart from this, the cold stress also reduces the chlorophyll content as well as that of all these related parameter of the photosynthesis, and it was reported in the eggplant, that is Solanum melangina, as well as the quantum efficiency of photosystem 2, maximum quantum efficiency, photochemical quenching, potential photochemical efficiency, and excitation capture efficiency of open center. And in this, the EBL application helps to elevate the inhibition of photosynthesis, which is caused due to the cold stress. Then we have a low temperature stress, that is a chilling stress, which affect the photosynthetic apparatus by disturbing thylakoid electron transport, the carbon reduction cycle, the regulation of carbon dioxide supply. The chlorophyll A and B content in plants decrease when subjected to cold treatment, and the brassinosteroids are capable to prevent the loss of these. As the various concentration of brassinosteroids have been applied, that is the 1, 0.1, 0.2, 0.4, so on, and it has been reported that the lower concentration of the AP brassinosteroid are supposed to overcome the effect of the cold stress. EBL improved the cold tolerance in the eggplant by regulating the photosynthesis. It was also reported. And then we have another BR27 that provided a defense against membrane leakage and the photosynthetic pigment degradation. EBL also induced the snow mold resistance in the sickle cereal cultivars, whereas the exogenous EBL application resulted in the lower total soluble carbohydrate as well as the enhanced photosynthetic efficiency in the other graph. Then we have a heavy metal stress that, as you know, that the metal up to a particular density are classified as a heavy metal. So, but the toxicity of the toxicity effect and symptoms produced by the heavy metals are minimized by the brassinosteroids, and one of the such study is related to that of the cadmium. As you know that the cadmium has the capability to decrease the activity of the chlorophyll reductase, chlorophyll activity, degrade the total chlorophyll content, as well as the reduce spared chlorophyll value, as well as the various photosynthetic and the other enzymes. It was reported in the various plants, so it is not possible in the short time period to describe each and every plant. So, but the major effect of the cadmium is that it decreases the photosynthetic efficiency overall, and the epibrosinolide has the capability to inhibit the effect of BR at a certain level, but at a higher concentration, the effect of uh, cadmium cannot be neutralized. This was also reported in the Lycopersicon esculentum in our laboratory. And we have applied the brassinosteroid with that of the cadmium itself. Then a PhD work was done on the nickel and brassinosteroids under the supervision of Professor Qazi Fariduddin, and we have got a similar result on this also, that a particular nickel concentration, a reduction in the chlorophyll and the chlorophyll fluorescence was noted, whereas when we are going to treat it with the HBl, it helped to repair the damage that has been caused by the nickel exposure in these parameters. Then the similar results were also reported in the case of the copper, and a number of crop has been studied in our laboratory also, and these are some of the results which we are summarizes. And besides that, there is a chromium which is basically here in the case are that contamination. Its contamination is due to the industrial use. It impacted the overall photosynthetic pigments and the photosynthesis system to quantum yield in the radish seedling, it was reported. But the EBL, which has the capability to mitigate here also, it mitigates the toxic effect of the chromium itself. And we have also the some effect in relation to that of the aluminum. Then we have the lead and mercury, as well as that of the calcium nitrate. So here we are just having a complete picture that what are the actually the phenomena which has been related to that of the various stresses, that I have already told you that the effect of the salinity, drought, heavy metal, temperature, it might be low or high, as well as that of the various other chemicals such as the various pesticides. So here you just see that the perception of the signal is there, and then that relay of signal which ultimately leads to the changes in the cell cycle, cell division, then the modification. Then we have a physiological and biochemical level where the changes are occur, and also at the functional gene level there is a response is created where the various enzyme transporter and stress nuclear genes were affected. Then there is a direct signal that where the receptors had received the signal, and these as transduces into that of the sensor, and the kinase phosphatase are involved. Then we have a regulatory role of these factors that is related to that of the transcription factor, post-translational level where the mRNAs are the engineering, genetic engineering are changing. So, and all these changes or decrease can be overcome by the application of the brassinosteroids. Then we have the mechanism that is related to that of the photosynthetic. So it will be already more. So it is just it is not directly related, but we can say that there is a brassinosteroid mediated regulation of the photosynthesis, which has been summarized here, and in little picture is little bit not clear, but here that it is a BR inactivation, and if there is a low BR, and then we have some of the effect which affect the chlorophyll biosynthesis BR, and ultimately it has to affect the various different enzymes that has been directly or indirectly related to that of the brassinosteroid biosynthesis. Finally, we have to just conclude that brassinosteroid regulate both photosynthesis as well as that of the photomorphogenesis in the plants itself. They promote the cell elongation, which aids the hypocotyl in emerging through the soil. Upon exposure to light, photomorphogenesis is triggered, leading to the development of the chloroplast and stomata, ultimately enhancing the photosynthesis. BR increases the stomatal conductance, facilitate the carbon dioxide intake. They also improve the light capturing efficiency of plant and increases the chlorophyll content, contributing to a higher net photosynthetic rate. So we can summarize that BR is one of the new class of plant hormone which has the capability to mitigate the effect of various stresses by inducing the various physiological process in one way or the other way by increasing the various physiological parameters. Thank you so much. Is there any question? If you want, you can ask. Now we have a short of time. Actually, it was supposed to be finished at 1:30, but it was now 1:54. So now we have to move to the lunch, and after the lunch, we have a tutorial session that is related to that of the PAM. So try to finish your lunch as early as possible.
In today's tutorial, we are going to learn about junior PAM, which is also known as pulse amplitude modulated fluorometer. So, before using this particular instrument, we should know its working principle in brief. So, basically, junior PAM is a pulse amplitude modulated or PAM chlorophyll fluorometry along with saturating pulse analysis of fluorescence quenching has become a standard method for evaluating various photosynthetic parameters. In the 1990s, the world's technique PAM or PAM 200 was introduced this technology in the university classroom. Junior PAM measures chlorophyll fluorescence to evaluate photosynthetic performance, particularly the efficiency of photosystem 2 or PS2 or we can say PS680. It is widely used in plant physiology for detecting plant stress and evaluating various photosynthetic parameters. Chlorophyll A, as we all know that chlorophyll A is very important molecule in photosynthesis. So, chlorophyll A fluorescent kinetics is widely used as a method for studying photosynthesis in plants, algae and cyanobacteria, which offers both qualitative and quantitative information. Various techniques such as PAM fluorometry method analyze fluorescence changes during photosynthesis, which basically measures rapid rising and slow declining phase in dark adapted samples or when they are exposed to light. The fluorescence induction curve, or we can say OGIP transient, plays a very important role in measuring chlorophyll fluorescence. So, basically it comprises of label O, which we can say as origin, Ji, which are the intermediate levels, and then is P, which is peak. These changes are inductive of the reduction of photosynthetic electron acceptors. The fast phase of this chlorophyll A fluorescence induction, or we can say OGIP transients, is widely studied due to ease of analysis and compared to a slow phase. So, basically this phenomenon is closely linked with photosystem 2 or we can say PS680, where variable is chlorophyll A, where variable is basically chlorophyll A fluorescence, which originates from photosystem 2 and it is influenced by electron acceptor sites. So, here plastoquinone or we can say QA act as a quencher. When it is in its oxidized stage, plastoquinone basically act as a quencher. So, the fluorescent rise reflects the reduction of quencher A or we can say plastoquinone A. Whenever there is rise in fluorescence, then plastoquinone reduced itself, and this will basically affect the redox reactions and there will be a photosynthetic electron transport chain. So, now what happens in OGIP? The OGIP transient now provides valuable insights into photosystem 2 photochemical quenching and electron transport activity, and it also affects various inhibitors, strators and mutations. Additionally, several factors such as photosystem 2 connectivity, P680 oxidation, pheophytin, plastoquinone pool and non-photochemical quenching and electric field can also modulate fluorescence. So, basically these are various parameters which can change fluorescence accordingly. We are going to take two samples, one is dark adapted sample and another one is light adapted sample. So, basically what happens in the dark adapted samples, here PS2 reaction center remains open. So, in dark adapted samples, PS2 always remains open, which results in minimal fluorescence, which is known as F0, and it is represented by O. Here O stands for origin, as we have explained in OGIP curve previously. The rapid OJ rise is driven by photochemical process and it strongly depends on light intensity, while the slower Ji and IP phase are associated with thermal processes. Under saturating light, when we are keeping our sample in dark light condition, then their fluorescence reaches its peak and we name it as Fm or F max. When all the plastoquinone molecules are reduced, we are giving our sample a light, then all plastoquinone molecules are reduced, and this will close photosystem 2. In dark adapted condition, photosystem 2 was open, while when all quinone molecules are oxidized, then photosystem will remain closed. Then a lower light intensities will change fluorescence little bit. Now notably, analyzing fast fluorescent induction curve under saturating light using parameters like F0 and Fm provides the critical information about photosynthetic efficiency and activity. So we are going to take F0 and Fm. Basically, these will provide us insights that whether there is change in photosynthesis or not, or whether PS2 activity is working properly under stress condition or not. So moving forward, as I have told you earlier, that we are going to work on two samples. First one is dark adapted sample, and another one is light adapted sample. So basically, in dark adapted samples, minimal fluorescence or F0 and maximal fluorescence, which is Fm, we are going to study them, and we are basically going to study whether there is change in it or not, and how much change we are seeing in healthy, stressful conditions. Then we are going to study a ratio of Fm and F0. Basically, ratio of Fm and F0 remains constant under different physiological condition, but only under non-stress condition or when our plant is healthy. But when we are giving any hormone treatment or when we are subjecting our plant to any stress, then Fm upon F0 ratio do change. The difference between Fm and F0 is referred to as variable fluorescence or FV, and FV by Fm ratio is commonly range between 0.78 to 0.84 in higher plants. So this is the range which is provided, that FV by Fm ratio should be 0.78 to 0.84 in higher plants. Basically, this is the indicator of maximum quantum yield of PS2 photochemistry. Now, another useful parameter, that is relative variable fluorescence, which is particularly at time t. So it is defined as Vt, and it equals to Ft minus F0 upon Fm minus F0, which normalizes the fluorescence induction curve for comparative analysis in various parameters, whether we are taking control or hormone treated or stressful plant. So moving next to a plant, when the sample is subjected to light, then what happens? So basically here, whenever the plant is light adapted, then there is change in PS2 activity, or PS2 regulates itself. However, exposure to intense light pulse results in temporary fluorescence maximum, which is F dash max, which is generally lower than Fm. Obviously, F dash m will be lower to Fm. We are going to take out Fm where? In dark adapted sample, and in light adapted sample, F dash m will always be lower than Fm. Non-photochemical quenching, or we can say NPQ, is quantified by the expression of Fm minus F dash m upon F dash m, and comprises of three components. Basically, NPQ has three components. One is QE, which is also known as an energy dependent quenching. Then is QT, which is state transition quenching, and then is QI, which is photo inhibition related quenching. So here, energy dependent quenching component is rapidly reversible and associated with thylakoid membrane, and delta PH, PSBS protein and zeaxanthin, while QT results from reversible photophosphorylation of light harvesting complex 2 or PS2. So these are various parameters which we are going to study in light adapted samples, that are NPQ, QE, QT, F0 by Fm, various parameters you will study here. Then you can compare your samples and you can say that what are the changes you are seeing in control condition, in hormone treated plant and stressed plant. So dark adapted samples are commonly used as reference point to a normalized data from light adapted samples. We are always using dark adapted samples as our reference point, and However, light adapted samples, we are going to sample them and note their readings, aiding its derivation parameters like Qn and Qp. So this is the junior PAM. As you can see here, we are going to demonstrate it in detail. So what are its application? Basically, junior PAM is used to monitor whether plant is in stress, that is drought, salt or temperature extremities. What are the photosynthetic change that we can measure by using this junior PAM, and how we can use this in our crop improvement researches and studying the impact of environmental changes on plant health. Now, advantages of this junior PAM, as you can see that we can measure here in real time measurement. We can measure here and we can take reading from here only. Secondly, it detects early photosynthetic alterations. As we know that photosynthesis is divided into two major components, that is light reaction and dark reaction. So early photosynthetic alteration we can study here while using this junior PAM. Suitable for different plant types and environment, we can study on different plant varieties or different plant species, and we can study it in different environments also. It provides detailed information on photochemical and non-photochemical processes. Apart from that, it is a compact device and it can be easily used in field studies. So this is a brief about junior PAM. So now I am going to tell you that how to use it. So basically, this is the junior PAM. It's too handy as you can see it, and I have connected it with this laptop. In this laptop, we are already having its software, and so I have connected this junior PAM with this laptop, and we are having here this very delicate fiber, and we have connected this delicate fiber here. Its one end is connected to junior PAM device and another end is connected to its sensor. These are magnetic clip, or we can say magnetic sensors. It will sense that what are the changes we are going to see. Basically, this magnetic sensor is used to study, or is used for studying of dark adapted sample, while this clip is used to study samples which are light adapted. So I am going to demonstrate you that how we can study it. Here we are having this, we can also take out this fiber from it. Okay. Very meticulously, you just have to close it. Now, what are dark adapted sample and what are light adapted sample? Basically, we have covered our leaves 30 or 45 hours prior to experimentation. We have to cover our leaves with foil paper. So when we are covering our leaf with foil paper, then this leaf is getting no light, means it is dark adapted. It is starving. Here photosystem 2 is open. So whenever we are going to remove this foil and light will fall on it, means the PS2 will activate itself and plastoquinones quenching will start to its peak. So we are going to get FM of F max here when our sample is dark adapted. Okay. So how to take its reading? Firstly, we have to remove, firstly in this laptop, let me show you. Yeah, these are tomato plants. We have to select all the desired parameters which we are going to take. Suppose I am selecting all parameters. Suppose we are going to take QP, QN, QL, NPQ, Y, F naught, FM, and FP by FM. This particular set is already calibrated. These are tomato plants. This is hormone treated plus stress. This is control one, and this is stress. Now we are going to study that what alteration we are going to get. So firstly, we'll study in control set. So basically in control set, we are going to remove this foil, and gently we are going to put this clip. It should be flat like this. Then here we are going to read our reading by clicking on set. So whenever we are going to click on set, then we are going to get here readings of different parameter. As you can see, FV by FM and FM. No, another parameters are not included here because this sample is dark adapted. Okay. So here we are getting reading of FV by FM as 0.375. It's too low because we are not getting light here, enough light. When we are working on field, then this reading would be, I think, in range of 0.765. But we are not getting enough light, so we are getting here reading of 0.375. Is it clear? Now we can move to another sample, but firstly you have to take it out very gently and close it. Now moving to next sample, which is our stressed plants. Very carefully you have to put this magnetic clips in between and place leaf in between. Now again same procedure, we are going to click on set, and again here we are getting readings of 0.211. So this means in control plants our FV by FM is 0.375, while in stressed condition it changed to 0.214. So FV by FM values are declining. Now moving forward to hormone treated plants, as these plants are having dust and dirt particles, this should be clean. Neither your readings will. Now this plant is hormone treated. So what are you thinking, that FV by FM value should increase? Then this control set. So let us see. Again we are going to remove this foil from our leaf. And then here we are going to click on set. You can clearly see that in hormone treated, FV by FM is 0.736. So it is clearly seen that in control set, FV by FM is 0.3, here 0.1 and here 0.736. So these are samples which are dark adapted. Now moving to light adapted samples. So what we have to do, we have to just take it out, clean it, then we have to move it, take it out, take our fiber out from it. Then we have to insert this fiber into this clip. This clip is basically used when we are going to study light adapted samples, and this particular magnetic one is for light adapted samples only. We have we can take any leaf starting from hormone treated only. Now here we can take different form of reading. Suppose I am going to take F0 by Fm. Then whenever I am clicking on F0 by Fm, only I will get reading which is F0 by Fm, but when I am clicking on set, then I will get reading which is QP, QN, QL and NPQ. So firstly, by clicking on F0 by Fm, so here by clicking on F0 by Fm, Fe by Fm value is 0.714. So why it is lesser than dark adapted Fe by Fm? Whenever plant is in hunger, so all the PS2 is open already, so all the light will strike on it, and basically all plastoquinone will start getting quenching. So we will get Fe by Fm always more in dark adapted sample than in light adapted sample. So here we are getting 0.714, which is hormone treated plant. We can also take set here. By set light, we can get different readings such as, here we can get different readings such as QP, QN, QL, NPQ. It totally depends on you that whatever reading you have to take. Suppose if you only have to take Fe by Fm, then no need to go for this all. But if you want to take QN, QL, then you can take by using this. Now moving to next, which is stressed plant. As you can see that morphology of this plant is already here. Here we are getting 0.277. Reading is 0.277. Obviously, Fe by Fm is lower than hormone treated plant. Fine. So you can compare here that Fe by Fm declined in stress treated plants. Now, if we talk about control set, then in control set we are getting Fe by Fm reading 0.632. Again 0.632 is lower than hormone treated plant. Apart from that, NPQ is 0.013, maximum yield which is Y is 0.500, QN is 0.021. So if you want to incorporate these readings in your data, then you can. It's totally depend on you that whatever readings you are going to take. Apart from this, we can also see light curves here. If you want, then you can go for it, but here you can easily see that in report we are getting all these kind of readings, which is on this side we are getting Fe by Fm, and then Fm, then F0, NPQ. So we are getting different readings. Whatever you want, you can work on it. Fine. Or again I should explain. Is it clear? So it's I think it's very simple one. Apart from that, you can save your data very simply. As you save data in MS Word. Just you have to click here on file and then save this data on desktop. It's quite easy. Now you all can come in 10, 10, group of 10, 10 and ये वही है जो रीडिंग वो ग्राफ तुम लोग को अपने डेटा में थोड़ी करना है, ये नया पैम है ना इसलिए ये नया है, इसमें तो तुम रिपोर्ट दे सकते रिजल्ट यहाँ से कर्व करते हैं। हटो कम आएगी, तुमने खाना नहीं खाया है, तुमने खाना नहीं खाया है तो क्या होगा?
lecture, and we must appreciate his stamina. And again, he is going to deliver on a very interesting topic, methods for modeling phytohormones mediated signaling in plant development.
Thank you very much. Hello again. Now, okay, oh, thank you. And now I would like to present how phytohormones regulate. We know that, of course, regulate plant growth, but why modeling this signaling pathways is essential. And then different approaches used to study them. Before diving into the details of modeling phytohormone signaling, let's first understand why phytohormones are so important in plants development. Of course, we know about not only my lectures that are chemical messengers that regulate almost every aspect of plant growth and development. They control key processes, and these hormones work in a coordinated manner and of course coordinated or interact with other phytohormones. But why should we model phytohormone signaling? There are two primary reasons. The first, of course, understanding complex pathways, and the second, predicting outcomes of genetics or environmental changes. Phytohormone signaling is highly complex, involving multiple feedback loops, interactions between different phytohormones, and responses that vary depending on the developmental stage or environmental conditions. By modeling these pathways, we can systematically analyze and predict how phytohormone regulate plant growth. What about the predictive? Computational models allow us to stimulate the effects of genetic modification, such as gene knockout or overexpression, or environmental factors too, such as drought or extremely low or high temperatures. This is particularly valuable for optimizing crop traits and developing stress-resistant plant varieties. Let's explore why modeling this signaling is crucial. We know that phytohormone signaling is not a linear process. It's a highly intricate network that involves multiple feedback loops that regulate hormone levels and responses. The second is a crosstalk between different phytohormones influencing multiple development processes. And the last is context-dependent signaling, meaning that the same hormones can have different effects depending on tissue type, developmental stage, or environmental conditions. Because of this complexity, traditional experimental approaches alone are often insufficient to fully map out hormone interactions. This is where a computational model becomes a powerful tool. By developing mathematical and computational models, we can systematically analyze how different components of hormone signaling interact. The second, identify key regulators that drive specific development or stress responsive outcomes, and integrate data from multiple experiments to form a comprehensive picture of how phytohormone function. Example, in auxin-cytokine interactions, computational model help us understand how hormone gradients influence root and shoot development. It's a typical physiological interaction between auxin and cytokine. It's typical even books. So, by stimulating this dynamics, we gain insight that would be difficult or even impossible to obtain through experiments alone. Ultimately, modeling allows us to make a prediction, test hypotheses, and refine our understanding of phytohormone signaling, all of which contribute to innovation in crop improvement, stress resilience, and prediction agriculture. Plants adapt to drought, temperature shift, and stress factors. Here we know why biotechnology enables genetics modification for improvement traits like drought resistance or enhancement yield. Computational model allows us to stimulate these conditions and predict how changes in hormone signaling affect plant development. For example, ABA signaling models predict stomatal responses to water stress, aiding in the development of drought-resistant crops. And the second, auxin transport models help optimize root growth and shoot branching patterns for better plant architecture. These predictions are sometimes save times and cost by reducing trial and error in experiments and improve crop trace by identifying optimal genetics modification and support precision breeding, enhancing hormone-driven plant responses. By integrating computational models with experimental data, we can design more and more resistant and productive crops. Now that we understand why modeling phytohormone signaling is crucial, let's focus on a specific example, brassinosteroids. I don't present the signal transduction of brassinosteroids in plants, but I would like to tell about modeling. Modeling brassinosteroid signaling help us understand how brassinosteroid receptor perceive signals and active downstream genes, and how brassinosteroids interacts with other phytohormones like auxin, gibberellins, cytokines, and others to coordinate plant growth, how brassinosteroid levels influence stress tolerance, especially against environmental challenges like drought or extreme temperatures. By stimulating brassinosteroid signaling networks, we can predict how changes in brassinosteroid activity affect plant traits, aiding in crop improvement. Why modeling phytohormone signaling network provides value insight, it comes with significant challenges. These signaling networks are highly complex, dynamic, and interconnected, made it accurate prediction difficult. Some key challenges are feedback loops and crosstalk, spatial and temporal variability, and limited experimental data. Many models require precision biochemical parameters that are difficult to obtain. So, addressing these challenges required advanced computational approaches and integration of high-resolution experimental data. Despite these difficulties, modeling remains a powerful tool for unraveling phytohormone function. One of the biggest challenges in modeling phytohormone signaling is its complexity. These pathways involve numerous genes, proteins, and feedback mechanisms, creating in a highly dynamic system that is difficult to map and predict. The key complexities are following: multiple layers of regulation. That means that hormone activity is controlled at transcriptional, translational, and post-translational levels. The second, non-linear interactions. That means the effects of one hormone often depend on the presence of others, leading to unpredictable signaling outcomes. And the last, crosstalk between pathways. For example, auxin and cytokinins interact during organogenesis, making it difficult to isolate their individual roles. Because these factors, traditional experimental methods alone, cannot fully capture the system behavior, mathematical and computational models help simplify and stimulate this interaction, providing a clearer understanding of plant growth regulation. Another major challenge in modeling signaling is feedback regulation and crosstalk between different hormones. Feedback loops can be positive, amplifying the response, or negative, that means suppressing the response, which tune fine hormone action. For example, auxin transport is regulated by pin proteins, whose localization is influenced by auxin itself, creating a self-reinforcing loop. And the next, crosstalk occurs when different hormones influence each other's pathways. For example, abscisic acid and salicylic acid exhibit antagonistic crosstalk, where abscisic acid position draw response, while suppression salicylic acid driven defense mechanism. This interaction introduces non-linear and context dependence, making it difficult to isolate the effects of a single hormone, I said. Once again, the same hormone can have different effects depending on its concentration, distribution, and developmental stage of a plant. We know everything about it. Key challenges include hormone gradients, tissue-specific responses, and time-dependent signaling. Hormone gradients, for example, auxin form localized maxima in root tips, directing meristematic activity and organ patterning. For the second, a hormone may promote growth in one tissue but inhibit it in others. Some hormones responses occur immediately, while others require long-term regulation. We know about these responses when you treated exogenously plants by phytohormones. To model these dynamics, Researchers use special modeling approaches, such as partial differences equation is PDF as an abbreviation, which simulate hormone diffusion and distribution in plant tissue. These models are crucial for understanding how hormone coordinate growth across different parts. The next challenge in modeling is the lack of precise experimental data, which need to define model parameters accurately. Many models rely on quantitative values such as reaction rate for hormone biosynthesis and degradation, hormone transport coefficient, and binding efficient between receptor and signaling molecules. However, these values are often difficult to measure directly due to, firstly, hormone instability and low concentration making detection challenging. The next, tissue-specific variability is required high-resolution techniques. And the last, complexity of interactions where multiple phytohormone influence each other dynamically. To improve parameters estimation, we are now integrating high throughout omics and data and advances imaging techniques to obtain more precise measurements. There are three main methods: experimental approaches, computational modeling, and integrative frameworks. The first, this include gene knockout studies, biochemical assays, and imaging techniques to observe hormone activity directly. In computational modeling, the mathematical and machine learning models help simulate hormone interactions, transport, and regulatory networks. At the last models, integrative frameworks, this combine experimental data with computational tools, creating a more and more complete picture of phytohormone dynamics. Of course, each model has its strengths and limitations, but together they allow researchers to better understand and predict plant hormone behavior. Experimental methods play a crucial role, we know, but key techniques include genetical studies, biochemical assay, imaging techniques. Of course, each of these methods provides valuable insight, but they also have limitation. For instance, genetics modification may trigger compensatory mechanism, making it difficult to isolate specific effects. This is why integration experimental data with computational model is essential for building a complete picture of hormone signaling. Genetic studies are one of the most effective ways to understand phytohormone function by manipulating genes which are involved in hormone biosynthesis, transport, and signaling. There are some key approaches: gene knockouts and gene overexpression. The first, removing specific genes using techniques like CRISPR-Cas9, helps identify their role in hormone regulating. And gene overexpression, by increasing the expression of key genes, involve us to observe their effects on plant growth and development. For example, Knockout studies of PIN transporters have revealed how auxin movement shape plant architecture. Similarly, overexpression cytokine-related genes has provided insight into shoot branching and meristem activity. While these approaches are powerful, they may not fully capture redundant or compensatory mechanism within signaling networks. This is why combining genetic studies with biochemical imaging techniques provides a more comprehensive understanding of phytohormone signaling. Biochemical assays are essential for measuring phytohormones level. We know about it. One of the most, of course, useful techniques which I presented today is LCMS-MS-MS, which provides high sensitivity and specificity, quantification of hormone fluctuation, etc., etc. We know everything from my previous lecture. Imaging techniques are powerful tools for visualizing hormone distribution and dynamics in plant tissues. These methods help us track where phytohormones accumulate, providing critical insight into their role in growth and development. One widely used approach is GFP tagging, where fluorescent markers are attached to hormone-related proteins or reporters. For example, DR5-GFP constructs allow real-time tracking of auxin maxima in root and shoot meristems. The next, fluorescence lifetime microscopy enables dynamic monitoring of hormone receptor interactions. While experimental approach provide direct insight into hormone signaling, computational modeling allow us to simulate, predict, and analyze complex hormone interactions that are difficult to study through experimental alone. Computational model use mathematical equation to represent biochemical reactions involved in hormone synthesis and degradation, signaling kinetics that regulate plant responses, and hormone transport and feedback loops, ensuring proper distribution and function. These models help identify critical control points in a signaling network and predict how genetics modification or environmental changes affect plant growth. However, computational modeling requires precise experimental data for validation. Integrating biochemical, genetic, and imaging data improves model accuracy, making it a powerful tool for crop optimization and stress adaptation research. Computational modeling of phytohormone signaling relies on various techniques to stimulate biochemical processes, spatial distribution, and regulatory interactions. There are four main approaches: mathematical and spatial modeling, machine learning and artificial intelligence techniques, agent-based modeling, and network-based modeling. The first technique use ordinary differential equation and partial differential equation to stimulate hormone synthesis, degradation, and movement across tissues. The second, machine learning and AI techniques, predict gene hormone interactions and signaling patterns using data-driven models such as neural networks. Agent-based modeling simulates individual cell behavior and how they collectively form tissues structures. Network-based modeling visualizes complex hormone, protein, and gene regulatory network, revealing key interactions in signaling pathways. What about ordinary different equations in mathematical modeling? Used to model hormone synthesis, degradation, and signaling kinetics over time. For example, ODE can predict how auxin levels or other, of course, hormones fluctuate in response to environmental stresses. Partial differential equation used for spatial modeling incorporating hormone diffusion, tissue-specific localization, and gradient formation. This approach-based models help explain how phytohormone maxima guide root and shoot patterning. These models are essential for understanding how hormone concentration change in time and space, helping us predict growth patterns, stress responses, and developmental decision. Machine learning and AI techniques are enable data-driven prediction of gene hormone interaction signaling dynamics, prediction hormone regulated gene expression. The next application is a pattern recognition in a large data set. The next, optimizing crop traits. What's the mean? Predicting hormone regulating gene expression. Deep learning models analyze transcriptional data to identify hormone response genes. In the second pattern recognition in large data set, neutral networks detect hidden correlation between hormones, environmental stimuli, and growth patterns. The next, of course, AI helps predict how genetics modification affect hormone pathways, guiding precision breeding strategies. For example, machine learning has been used to predict auxin distribution in root under varying gravitational conditions, improving our understanding of gravitropism and plant adaptation. Despite its power, AI models often lack interpretability, requiring biological validation through experiments and traditional computational methods. The next, agent-based modeling, is a powerful technique for simulating how individual cells behave and interact in response to phytohormone signaling. Unlike traditional mathematical models, agent-based modeling treats each cell as an independent agent, responding to local hormone concentration and external stimuli. There are some advantages of this method. The first, capturing emergent behavior, modeling transport at a cellular level, and the last, studying experimental responses. Of course, by adjusting parameters, we can predict how plants respond to drought, light changes, or nutrient availability. This approach is particularly useful in modeling root meristem activity and shoot branching, helping researchers understand how hormones shape plant development. To fully understand hormone signaling, we must integrative frameworks which combine experimental data, computational modeling, and large-scale biological data sets. This approach provides a holistic view of how phytohormones regulate growth and development. There are some key components which integrate frameworks. Multi-omics approaches, integrating transcriptomics, proteomics, and metabolomics to reveal hormone-regulating pathways. I can say not only but plant hormonomics, but we know that's a part of metabolomics. Bioinformatic tools, for example, platforms like the Arabidopsis EFP browser help visual gene expression changes in response to hormone. And the last, multi-hormone integration modeling. Understanding, for example, interaction between two hormones, crosstalk in cell expression of this hormone interacts in stress responses. By merging experimental computational data, integrated frameworks provide a more accurate and predictive models of phytohormone function, benefiting both basic research and agricultural application. One well-studied example is the crosstalk between auxin and gibberellin, which regulates cell elongation, division, and overall plant growth. There are, in model, there are some interactions. Gibberellin enhances auxin response. That means that this hormone promotes the expression of auxin responsive gene, facilitates cell expansion. And then auxin regulates gibberellin biosynthesis. That means auxin stimulates the production of gibberellin in rapidly growing tissues, ensuring coordinated development. And the last, tissue-specific effects. In short, auxin and gibberellin interactions drive stem elongation, while in root they help regulate meristem activity. We know that abscisic acid and salicylic acid play opposing roles in plant stress responses, making their interactions a key area of study in phytohormone signaling. Abscisic acid promotes abiotic stress tolerance, so it helps plants survive environmental stresses, environmental conditions by regulating stomatal close and gene expression. Salicylic acid enhances biotic stress defense, so activates immune response against pathogens. Antagonistic regulation is under, for example, under drought conditions. Abscisic acid can suppress salicylic acid signaling to prioritize water conservation over pathogen defense. Conversely, during infection, salicylic acid reduces abscisic acid activity to enhance immune responses. What about next case, auxin signaling? Auxin transport models like canalization hypothesis explain how auxin flow directs patterning in leaves and root development. The next, of course, in auxin modeling Perception and signaling is important. I thought about receptor complex, mediates auxin responses, regulating transcriptional factors that drive growth. We know that important is gradient formation. PDE based models simulate how auxin maxima influence cell division and differentiation, especially in meristem. The key feedback mechanism include in this case positive and negative feedback loops. What's the mean that auxin promotes the localization of pin transporters, reinforcing its own directional flow, which is crucial for vein patterning and root development. But negative feedback loops mean that high auxin levels trigger degradation of auxin repressors, ensuring tie regulation of gene expression. Crosstalk of other hormones. Auxin interact with cytokinins or gibberellins, creating context-dependent growth responses. The next case of study, cytokine signaling. There are two key aspects, two components signaling system, such as cytokinin receptor, for example, CRE1, activate histidine kinase, which transfer phosphate signal to response regulators. The next is should and root development. Computational models simulate how cytokinin gradients influence meristem activity and branching patterns. To fully understand phytohormone regulation, we may use multi-scale modeling, integrative all data from different biological studies, molecule, cellular, organismal levels. The first models focus on gene expression, protein interactions, and hormone biosynthesis. Cellular level mean that simulation track hormone transport, signaling cascades, and feedback loops within individual cells. What about organismal level? Models predict how phytohormone ingredients shape root growth, shoot branching, and stress responses. This approach bridges the gap between small-scale molecular interactions and the large-scale developmental patterns, helping us simulate plant responses under various conditions. What happens at the molecular level? Phytohormone bind to receptor, triggering a cascade of biochemical reactions. Transcriptional factors are activated, switching specific genes on or off to control plant responses. And enzymes synthesize, modify, and degrade phytohormones, adjusting their level based on the plant needs. Why model it? To simulate how hormone receptor interactions drive gene expression, to predict the effects of mutation on signaling pathways, and to understand how environmental factors influence phytohormone biosynthesis. By manipulating these molecular interactions, we can design experiments, optimize genetic modification, and develop stress-resilient plants. What about on the cellular level? What happened inside a cell? Hormones, we know that are synthesized and transported. Some move freely, while other phytohormone required carrier proteins. Signaling cascade begin when hormone, of course, bind to a receptor, serial molecular signals spread through the cell, and feedback loops regulate the process, ensuring hormone levels stay precise and balanced. Why why model the cellular level? To simulate how hormones flow through individual cells and predict their interact on development, to understand how cells respond to fluctuation of hormone levels under stress or normal, of course, conditions, to study localized hormone distribution, how proteins, for example, a direct auxin flow, a pin protein in root or system tissue. From individual cells, let's zoom how phytohormone influence entire organ and coordinate plant growth. At this level, phytohormones act as global messengers coordinating all phytohormones, coordinating growth across different tissues and organs. What happened at the organismal level? Hormones create gradients directing root and shoot development. Environmental signal like light changes or draw trigger system-wide hormone responses. And hormones interact to balance growth and defense, ensuring the plant adapt to stress. Why model the whole plant? To simulate how hormone network shape over plant architecture, to predict how genetics modification impact plant form and function, and to optimize crop traits like improving drought resistance, for example, through abscisic acid regulation. Now that we have covered molecular, cellular, and organismal level, let's discuss about challenges bridging these scales in infinite model. Oops. What makes multi-scale modeling difficult? The first, different scale requires different data types, gene expression data at the molecular level, transport dynamic at the cellular level, and growth pattern at the organismal level, on 3 levels. Processes at the level affects higher levels, up predictable. For example, a small mutation in a hormone receptor can alter whole plant architecture. Computational complexity, simulating entire plants with high accuracy demands massive data sets and processing power. How tackle these challenges? For example, to create more accurate predictive models of plant growth, or to optimize plant traits for agriculture, improving resistance and productivity, or to design experimental efficiency, reducing the need for costly trial and error in genetical modification. So, despite the difficulties, advances in big data, AI, and high-resolution imaging are making multi-scale modeling more powerful than ever. Creating a model is just the first step. This is the real challenge which improves that it works. validation is crucial to ensure that our prediction align with a real biological data. How do we validate models? By comparison with experimental data, by sensitivity analysis. We need key parameters, for example, hormone concentration, reactant rate, to see how much they affect the outcomes, and parameter estimation. That means we must have advanced techniques like Bayesian interference and machine learning, which help fine-tune models using real-world data set. Why is validation important? Because it increases confidence in model prediction, identify weakness, and ensures practical application. So, validated model can be used to design stress-resistant crop and optimize growth conditions. Model validation is an ongoing process. The more accurate the model, the more powerful its predictions. No model is useful unless it matches real-world observation. How is done? By simulated hormone gradients, by gene expression level predicted by models, and predicted hormone response, which are tested under controlled laboratory conditions. Why is it important? Because it's ensured that models accurately represent biological system. It defines this appearance that help refine models and builds trust in computational predictions, make them more useful for real application. Even the experimental validation model must be robust and adaptable. This is where sensitivity analysis play a key role. This is where sensitivity analysis come in. How does it work, of course? We need to understand which parameters have a biggest impact of our predictions in our models. How does it work? Individual parameters are systematically varied, so such as hormone production rates or receptor binding affinities, and model responses are observed, which changes have the biggest effect on plant growth. And the critical components are identified. It helps prioritize which biochemical processes need further environmental validation. Why is sensitivity analysis important? Because it shows which model assumptions are most influential. It helps refine experimental tools, reducing unnecessary lab work. It improves model reliability by testing its stability under different conditions. After, of course, identification key parameters, we need precise value of them. How do we estimate unknown parameters? It's important by using the least square regression, mathematical statistical analysis, fits the model to experimental data by minimizing the difference between prediction and observation. The second, Bayesian interference, use probability to refine parameters estimate based on prior knowledge and new data, and machine learning techniques, neural networks, and optimization algorithm which can improve model accuracy by analyzing large data sets. Why is parameter estimation important? Because fill in gaps where direct experimental measurements are impossible, improves model accuracy, making predictions more biologically relevant, and then prevalent overfitting, ensuring model work beyond the data set they are trained on. Even with the best model validation, it's never perfect. Next, we must know about the biggest challenges in model validation and how we can overcome them. It's very important. Imagine trying to predict. We know that weather sometimes often with the best technology. Imagine predicting the behavior of living, growing, adapting plants. That's the challenge, the next challenge of model validation. Why is it so hard? Because plants are unpredictable. Two plants with the same genetics can still behave directly due to the microenvironmental variation. Some data is missing. Scaling up is stopped. A model that works for a single cell may fall when applied to entire plants. How to improve validation? We must run models under multiple conditions to test their adaptability, use machine learning to fill in data gaps and refine predictions, and commonly compare with new experiments to adjust and improve accuracy. To the end of the day, models don't need to be perfect, but they must be useful, helping us understand plant better and development more resilient crops. Where is phytohormone modeling headed? The field of phytohormone modeling is evolving rapidly, driven by advancement in data science, imaging, and computational power. So, what's next? Integration with high throughput omics, real-time modeling with AI, advanced imaging techniques, for example, fluorescence lifetime imaging microscopy, and super resolution microscopy will provide unprecedented insight into hormone movement and interactions. Why does this matter? More accurate models will help us predict plant responses to climate changes. AI-driven modeling can optimize breeding strategies for stress-resistant crop. Multi-scape integration will allow precision agriculture, where models guide real-time decision-making for farmers. The future of phytohormones modeling is a fusion of biology, artificial intelligence, AI, and high-tech imaging, pushing us closer to a new area of precision plant science. Let's dive deeper into how AI is transforming real-time hormone modeling. Imagine if we could predict how a plant will grow before it even happened. That's the promise of AI-driven real-time modeling in phytohormone research. How does AI improve modeling? By instead predictions, AI can process massive data sets and predict how plant response will respond to the genetic or environmental changes in real time by pattern recognition. That means that deep learning models identify hidden correlation in hormone signaling that human might miss. And by adaptive simulation, unlike traditional models, AI can continuously update itself, refining prediction as a new data became available. What does this matter? Help optimize genetics modification to enhance plant traits, enable early stress detection, and improve precision agriculture, where hormone-based model guide crop management decision. As AI continues to advance, we are moving towards a future where plant growth can be simulated, optimized, and ever controlled with unprecedented precision. So, how advanced imaging technology is enhancing model accuracy? To improve the accuracy of phytohormone models, we need better data. And that where cutting-edge imaging technology come in. This tool allow us to see hormone in action, capturing the movement and the reaction with unprecedented detail. Key imaging transformation hormone modeling are fluorescence lifetime imaging microscopy, I said about it, confocal and super resolution microscopy, and biosensor of Lyme cell living. This is a new molecular probes allow scientists to measure hormone fluctuation in real time, improving model accuracy. What does this matter? More precise imaging, reduce error in computational models, help confirm how hormone move, interact, and respond to environmental changes, and enables new data-driven improves in hormone-based crop breeding and stress adaptation. By combining high-resolution imaging and AI and computational modeling, we are entering a new and precision plant science, where we can see, predict, and manipulate plant hormones dynamics with extremely accuracy. Thank you very much. 1 hour. Yeah. I'm tired. Thank you.
Thank you, Professor, for a nice lecture. That's great. 1 hour continuous. And today you have completed 3 lectures? 3 lectures आज में क्या जाए? Today 3 lectures is too much. So, okay, thank you, students. And tomorrow we have again the class at 9:30, especially for the students of Professor Qazi Fariduddin and Professor Shamsulyar. They come in time, 9:30. 9:30 means 9:30, okay? And all other are requested to please come in time, because it is not good that the teacher and presenter are here and the students are not coming in time, okay? Okay.